इंट्रोडक्शन दे देते हैं participants को.
Okay.
So good morning all the participants, and today I want to welcome Dr. Sandeep Kumar from NPPGR. He is the speaker for today's session. So we will start our lectures with his schedule. So I will share his brief biodata. I hope it's visible to all. Is it visible?
And presentation mode, yes, visible.
Okay. So, sir is presently posted as principal scientist in the division of genomic resources at ICAR and BPGR, and his field of specialization includes the molecular biology and biotechnology. Sir's salient achievement includes identification and registration of various weed germplasm, which are conferred for rust resistance. Then he has identified and registered many of the wheat germplasm. Mainly his work is on wheat. So on spot blotch resistant germplasm he has registered. Then drought tolerant wheat germplasm have been registered by Sir. Then evaluation of the entire Indian wheat germplasm collection for rust and spot blotch resistance. Then identification of the QTLs which are related to the spot blotch resistance. Various other traits like stay green, various drought-related other traits, and development of the core set for the entire northeastern rice collections also have been done by Sir. His one of the most important achievement includes the stem and rust resistance in wild relatives of the wheat with the genome that was established by Sir's contribution. He also has conferred many of the awards in his name, like R.S. Paroda Young Scientist Award, Young Scientist Associateship Award, Boycott Fellowship during 2008-9, then Italian Government Short-Term Fellowship also. He's the member of the expert working group on wheat phenotyping, working globally, headed by INRA France, the Society for Green World and Sustainable Environment, Society for Advancement of Agriculture Science, the Indian Society of Genetics and Plant Breeding, and various other societies. And his publication mainly includes in the area of mining of the Indian wheat germplasm collections for especially for leaf rust and spot blot resistance, then DNA fingerprinting and genetic diversity work. And one of the most accomplished work of Sir include evaluation of 19,460 wheat accessions which have been conserved in Indian National Gene Bank. And with this, he has identified various new sources of rust and spot blot resistance. So with this brief introduction, I would like to welcome Sir for today's session. Sir, now the session is over to you. I request you to kindly start the lecture. Thank you.
Thank you, Dr. Mamta, for describing my work, for describing me in such a nice way. I'm very much glad to be the part of this training program. The slides are visible?
Yes, sir, it's visible.
Okay, it's good. So my talk is on genome-wide association studies. Actually, since being in a NVPGR, we are very fortunate that we have lots of germplasm with us, and we can use it for the different purposes. We can use it for the identification of new genes. We can use it for the identification of new germplasm lines, which can be used in the breeding programs. So what is association mapping is? Association mapping is a method of mapping QTLs that exploit linkage disequilibrium to link phenotype observed characteristics to the genotype, the genetic constitution of an organism, discovering genetic association. It is generally performed by scanning the entire genome for looking into the significant association between a panel of SNPs and a particular phenotype. That phenotype is depend on what character you are using, you are phenotyping your population. The association must be then independently validated to check the either they are contributing to the trait of interest, so that the association which we identify that can be used in marker assisted selection. is a kind of journey from one place to another place, like so we have the different markers in between. There's number of markers there in the genome. These markers are nothing but these are also the genes or the DNA segments. And we have the a long journey, like as we start from Ludhiana to Delhi, it's kind of journey. And there are so many cities in between. Likewise, we have so many markers in between, the markers which we have identified to the the QTL, because there are so many markers, and if we have to reach and we have to land close to the QTL. So there are many things, many approaches which we follow for this purpose. This germplasm is the fuel for the engine, because we have lots of germplasm here in NVPGR, and our cultivated gene pool in almost in all the crops. This is limited, and we variability is very limited, and we need to broaden the the genetic base of this cultivated varieties. And for this purpose, we need the novel variability for the useful genes, for biotic, abiotic traits, for quality traits. We have to evaluate our germplasm lines, especially the landraces, which have not been exploited so far too much in breeding programs. And these landraces are really good reservoir for the different genes, very useful genes like Nephal, which has been exploited for glue D1. The double null traits, exceptionally low viscoelasticity for biscuit and wafer is there. Very recently, one of our landrace of wheat, Hengo 2, which was, which belongs to the Shimla region of India, and where we have identified LR80. So likewise, there are so many genes are there in the landraces, and we can identify, we can explore this germplasm using the different approaches, especially like the association mapping. Only thing is to integrate the molecular tools so that we can identify, we can characterize this germplasm in the best manner. As I told you, NVPGR, this is a repository of the diverse germplasm. We have a germplasm of wheat, a good collection. We also have a very good collection of rice. We have a very good collection of maize and other crops. This germplasm is lying in the gene bank. Majority of that has not been used in the breeding program, or neither for the breeding program nor for any molecular work. So the chances of getting novel QTLs or the genes or the germplasm lines which can directly be used in a breeding program is quite high. So we have to link it with our identification of these with these GWAS studies. So this is the need of our to broaden our genetic base of our major crops, to diversify the collection of uncharacterized germplasm. We have lots of germplasm with us. We can do the precise phenotyping, which can be associated with the precise genotyping using the different marker systems like SNP, SSRs. Then we can use the different tools like this TESEL, GAPIT for identification of the genes or QTLs. That genes which are conferring there, maybe that can be validated in an independent population before to be used in marker assisted selection. We have the two different approaches for the identification of the QTLs. One is linkage based analysis, where we use biparental population biparental mapping. Like we use two parents which are diverse in nature, which have the contrasting features. We make the crosses, develop RILs or NILs, and then we try to identify the QTL interval regions. That is good, but the thing is, this variation is limited to the parents, only two parents, which we use here, for developing the mapping population. This recombinant event is not, are very limited, so the resolution is not quite high in comparison to the genome-wide association studies, where we exploit the historical recombinant events. Because these germplasm lines what we use, they are not related to each other. They have lots of diversity. Their genetic base is different. So that historical recombination is there because there's a pass through number of recombination events. So this natural genetic diversity we can exploit for the identification of the QTLs, and the resolution, of course, that QTL which will identify in the different genetic backgrounds. I think we can at the same time, we can also validate it. We can also do the fine mapping, and that can be used maybe in the marker ancestor selection, because this is validated in the different genetic backgrounds. There are certain advantages of association mapping over biparental linkage mapping. There's no need to develop any mapping population. We can use the germplasm lines, accessions, breeding lines, and we can exploit the historical recombinations. There are greater allele numbers and the broader reference population is there. Possibility of exploiting historical measured traits is quite high. And the QTL is useful, there's lots of diversity. QTL useful in diverse genetic backgrounds, much higher mapping resolution is expected. Earlier, actually, there was an issue with the, how we can exploit these historical recombinations, because markers were very limited and we were depending upon the markers like PCR based markers. We could not apply, we were not in the position to apply number of markers at a time. But now, since we have this next generation sequencing and next generation based markers, chip based markers, so we can apply number of markers. So it is easy, quite easy for us now to do the GWAS studies. And this GWAS offers an effective and objective approach for better chance to identify disease-associated variants, improve understanding of disease etiology, improve ability to test gene into gene interaction, and predict genetic disease risk. So then again, the question is, what is GWAS? GWAS, actually what we do, we make some certain kind of associations between, the SNPs, single nucleotide polymorphism, and with a trait of concern. So we identify the genomic region which is associated with the phenotype, fit a statistical model at each SNP in genome, use fitted models to test null hypothesis, no association with SNP and the phenotype. So we use number of lines where variation is there. Variation for SNPs is there. And this SNP, single nucleotide polymorphism, we exploit for this purpose. We match it one-to-one with all the SNPs. We match with the phenotypic variation, whatever we have in the population. And we see which SNP is matching with that particular phenotypic variation. So for each SNP, we conduct a test of association with the trait. We see whether this SNP is matching or some other SNPs are matching. So each SNP we match, like thousands of the SNPs are there when we do the chip-based studies, like 30,000, 50,000, maybe 1 lakh, or maybe 4 or 5, 6 lakhs. Out of that, we identify only few which are polymorphic in nature, and we don't consider them SNPs which are monomorphic in nature. And then what we do is we identify only those which are having the good association with the trait of concern. So SNP has a direct biological function because we see the functional polymorphism, SNP in LD with functional polymorphism. So it's a kind of indirect association, because actually what we do, depends on the markers. It's a kind of disease. So we see how it is linking with the phenotypic variation. These markers are in the linkage disequilibrium with other SNPs, with other markers. So we consider only those markers which are showing some kind of association with the disease. As you can see here, marker and this association with the disease. So when we apply for this GWAS scheme, actually, there are many things which we do. We have the phenotyping. First, we collect the germplasm of our concern. We have the different germplasm lines, but before going for this, actually, we cannot perform GWAS and for all the lines. We have to identify a set of lines which is diverse in nature, and then this germplasm lines, we do the phenotyping of this. And when we do the phenotyping, we are very careful and consistent phenotyping characterization. We identify the samples which are diverse in nature. So we perform different kind of test that I will tell you in the later. And then we do the genotyping. For genotyping, we have the choice of the genotyping platforms. Commonly, this Illumina or Affymetrix. We have the choice of genotyping products like SNP density, We also do this DNA quantification and prediction. We do the data cleaning. So the data cleaning, what we do is samples genotyping has the is high if we remove the SNPs which have the minor alleles. For SNP genotyping success rate, like Hardy-Weinberg equilibrium, we also do. Heterozygosity, we also consider. Minor allele frequency, we also take care of. Then we do the in silico genotyping and the test for association and this replications. Here you can easily understand how we do this genome-wide association studies. We start with the field or lab greenhouse data. We have the phenotyping data, the diverse set of lines. Then we do this genotyping with the help of this next generation sequencing, platforms. Then we see the kinship or PCA, principal component analysis, to see the population structure, relatedness, and LD. We apply different statistical models like GLM, MLM, or CMLM for the association detection and validation. Then we perform this bioinformatics analysis to identify the QTLs and the candidate genes. And then we do the validation of these candidate genes before they're used in the marker-assisted selection. So I think you're not getting it complete. So we use a different marker system. As I told you, there's SNP data and the phenotype of different traits. So same panel which we genotype, maybe that can be used for number of purposes. For number of traits, we can evaluate that line, that set of lines, and we can identify the QTLs for number of traits, like multi-trait QTLs we can perform if we have the genotyping data of a set of lines. So after doing this association, we see whether this nucleotide polymorphism is associated with the phenotype or in the natural population. And after having this association, we exploit the extensive recombinations that are present in that map, in that set of lines or the natural population. So this, I have told you that this is SNP's single nucleotide polymorphism, which is present in our germplasm lines, because there's lots of variation is there, and we connect it with functional diversity, which is there in the form of phenotypic variation for different biotic, abiotic, and quality traits. So this purpose is to exploit the historical recombinations which is present in our germplasm lines, especially in the land races, because there's no breeding program has been taken place, and this material has not been exploited for any other purposes. So there are certain advantages I told you in case of biparental mapping population, or of the Jivas over biparental, because the resolution is quite high and there's no need to develop any mapping population. But there are also certain limitations, like heritability values is quite low in comparison to biparental mapping, and type 1 and type 2 error is there, means false positive associations are there in case of association mapping, so which we take care of by applying different tools like bone-Ferroni correction factors or FDR, false directive ratio. So these studies are now possible because we have sophisticated tools. We have NGS. We can do this SNP genotyping within few weeks or within few months. Earlier, it was taking years to do the this SNP, to do the genotyping, and that the genotyping was also not quite well, because we are not having that much of coverage as we can have with the help of these robotic systems. Number of this Jiva studies has been done after this decode of human genome has been decoded in 2007, and since then number of studies has been done. Association mapping study has been done, not only in human being but also in the plant genome. The problem with the Jivas is not having any caution, like suppose that genotype at a particular SNP are significantly associated with disease or with a trait. So this may be because of SNP is associated with some other factors. A confounding effect might be there, which is associated with the disease or with the trait of concern, but is not in the same causal pathway. So this kind of things we have to take care of while we are performing association mapping. So this association actually we perform, we apply number of markers. As I told you, we take care of this correlation and the linkage disequilibrium. And this is like phenotype, and we will apply the front of markers to see that, this genetic association between the phenotype and the trait of concern. Here, this trait of concern is disease susceptibility locus. Another thing which is really required for performing JIVAS is we must have sufficient sample size. As big, as large your sample size is, I think the success rate will be as high, because if you have a limited size of population, that the chances of genetic drift is there, and that you will have the skewed variation over there. So that we have to take care of while performing JIVAS studies. So there's a set of markers has to, contain a marker that is sufficiently correlated with the disease susceptibility loci, so that the genetic association at DSL is also visible at the marker locus. So linkage disequilibrium, we have to see how much it is. As much as linkage disequilibrium is there, chances of success is high. Genotype quality. So what markers you are using, how much false positive rate does not, dilute the real signals. So we have to take care of false positives. We have to take care of the false negatives also while we are performing this kind of studies. Linkage disequilibrium is affected by what population you are using. If you are using strictly self-pollinated crop, like wheat, chances of getting success is quite high in comparison to the crops like cross-pollinated or often cross-pollinated crops. So the linkage disequilibrium values would be lower. Another thing which affects the LD values is genetic drift. It depends on the population size. If population size is small, also the chances of a random effect is there, and obviously the LD values would be affected. Inbreeding will also affect the genetic drift. Mutation is also a factor which affects the genetic drift, this linkage disequilibrium and the gene flow. Because LD become larger if two population intermating are genetically distinct. Not much of a problem if crossing between highly similar populations found with most breeding programs. As I told you, in self-pollinated crops is good in comparison to lines where which are outcrossed. So methodology of AM. So as far as this methodology is concerned, we have to first define a population for analysis. So the population which we use should represent the diversity useful for goals of the project, specific to target of the project. Maybe if we are dealing with, say, for example, if we are evaluating the lines for leaf rust resistance, so there must be some lines which are highly resistant to leaf rust. Moderate level of resistance must be there in some lines. Moderate level of susceptibility must also be there. And there must be some lines which are susceptible or highly susceptible to leaf rust. Means a diverse array of diversity must be there in the set of lines which you are evaluating for association mapping purpose. Also, what you can do is you can use the lines from all major subdivisions of the species, or maybe you can take the lines from the different regionals or from the different localities, or maybe different states of India. So second thing is genotyping of the population. So genome-wide as markers, you use the the low density or low resolution like 100 SNPs. It all depends what is your purpose. or maybe you can also use a gel-based markers. So you can have only few markers at the time, or maybe you can have the medium density when you have the markers around 1500 SNPs. You can have the high density where you will use more than 50,000 to 5 lakh SNPs, or maybe you can have this like 2000 and 2 lakhs 50,000 SNPs in case of Affymetrix chip. Sometimes what we do, we also do the candidate gene association mapping, where you use a selected genes or selected set of SNPs, which we use for the purpose to just target some of the genes in our population. So SNP genotyping, what we do is this is the determination of SNP loci on a whole genome scale or within genomic regions of the interest. A large number of technologies or commercial platforms have been developed for SNP genotyping. Common approach for SNP genotyping include SNP arrays, tagman SNP genotyping, mass array SNP genotyping, and high throughput sequencing. So this is the region which we exploit for this purpose. This is the Affymetrix array. This is how it works. This is single nucleotide. You can see the variation over here. And second is Illumina-based bead array. Beads contain a sequence complementary to the sequence adjacent to the SNP loci when it binds to the DNA and exhibit different colors. This is about the tagman SNP genotyping. So different genotyping platforms are there. This is about a mass array SNP genotyping. So mass array platform has several advantages for the users who desire an accurate custom SNP genotyping assay. The steps in the genotyping using mass array, IPLEX system, locus specific amplification regions, SNP and SAP enzyme, neutralize uninterrupted DNTPs. IPLEX assay, the products are analyzed by the MALDI-TOF methods. We can analyze multiple SNP sites in one reaction and in accurate and cost-effective way. So cost will be much lower. There are different, this platform, next generation sequencing, this like Illumina or Roche 454 or Eintorrent. Imputation of data is important to see which marker is working and how effective, assessing a value of, something by interference from a value of the products or process to which it contributes. So we have to assess the value of a particular SNP so that we can see which SNP is giving higher p-value or which value is giving lower p-value, something like that. So these the different imputation methods which are there, like mesh and fast phase, spirit, different methods are there. Oh, right. Can I minimize it? I want to minimize it. Okay. Okay. Issues in genetic association studies, like many genes are there, like 25,000 genes, maybe the candidates. Many SNPs are there, like sometimes maybe more than 1 crores or maybe like SNPs are there. Are different methods are there to select SNPs. So to select only the functional SNPs in the candidate genes, systematic screening of SNPs in a candidate gene, systematic screening of SNPs in an entire pathway, or genome-wide screening, and systematic screening for all coding regions. So there are two sources for false positive detection, like positive in population structure, allele frequency differences among individuals due to local adaptation or diversifying selections, and familial relatedness or kinship. Sometimes population are linked, having some kind of relationship with each other that also affects this LD values. And if not, the popularly controlled can cause spurious association is there. So we have to take care of this kind of spurious associations because of Jivas, sometimes because of this false positive, sorry, you can find some spurious associations there. To how we can identify this false, discovery, this associations, so that different checks like FDR is there, false discovery rates. So expected proportion of false discoveries among all discoveries offers most powerful than Bunfunani, holds under weak dependence of the test. And there is Bennian approach, taking a priority into the account. False positive reports probability is there. So false positives means association is not there, but we see some associations there. So that we have to take care of. So Bonferroni correction is there. We set some limits in the program so that we can remove all those false positives. And we perform different tests. And also this permutation or recombinations we also perform, so that we can identify which combination is working perfectly for the association. And there is accounting for population structure or relatedness. So different subpopulations are there, and we take care of that, how much diversity is there. Sometimes, we don't get this normal distribution. So we remove the outliers also from the values, or we use this BLUP or blue values, so that when we deal with the data which has been collected from the different locations or which has been collected from the different years. So we have the different tools to evaluate the population structure, like principal component, so that we can see the fixed effect, define the groups of individuals. We can select the number of principal components that accounts for a specific amount of variation in the population. We also use the software, structure software, so that we can see how many subpopulations or admixture is there. We also evaluate the relatedness also, and also see the random effect. Sorry. Presentation. And as another statistical analysis, so we do marker by marker analysis. Like suppose if we are using, hello. If we do the statistical analysis, then we do go for this marker by marker analysis. And each marker, like say, for example, if we are using 35K FE matrix chip, then exomarriage chip, sorry. So what we do is all these 35,000 markers will match with the phenotypic value. So we do this marker by marker analysis. Analysis must be controlling for the population structure and also the relatedness. Bonferroni correction, as I told you, is the procedure to control the family-wise error rate. Probability of making one or more type 1 error. So there are two types of errors are there, type 1 and type 2. And it's the simplest and most conservative method to control this false detective ratios. And there is false discovery rate, FDR, is also the procedure to control the expected proportion of false discoveries. So when the association is not there, and we think that association is there, or sometimes what we see, there's association, but we are unable to identify that association. So in both the cases, we apply different correction factors like Bonferroni or FDR. So we use the different checks, like we said, p-values, how much p-value we want. You can say value we set before deciding which markers will you say that significant, or what p label should be, or whether it should be less than this or higher than that we decide. So if p value is 0.05 and if you have like 1536 SNPs, so it would be divided by this, and one for any value would be like 3.3 into 10 to the power minus 5. So as much as the markers would be, this bone ferroni will be lower. So this would be better if we apply more SNPs, or a chip which is having more SNPs in comparison to the other markers. Another approach is conservative approach, which we apply where we have this FDR, where we set this p value like less than 0.05. And there is selection of the best model. Which model would be applicable for us? So we see the expected value and the observed values. We match it with the expected values, the observed values. This is we call the QQ, quantile quantile plot axis, how well the model which we are using is fitting with the expected values for population structure and family relatedness. Negative logarithm of the p-value from the models fitted in the GWAS are plotted against the expected values under the null hypothesis of no association with the trait. This another thing which we do is Manhattan plot for where we see for each for chromosome wise distribution. You can see in all the 21 chromosome of wheat, and this is a cutoff value which is here. You can see this line, and the association over this line are the significant associations. And you can see there's some new loci which have been highlighted by red color, and there's also loci which are highlighted by the pink color, but they are not new. They have already been reported by some other persons. So this is what you can see. You can do is you can find the values which are much higher. Maybe this association is quite useful, or association where you are getting good peak. So a good quality Manhattan plot of the common variants show cluster of similar p-values. Neighboring variants supports each other. So this kind of associations we require, like this one, this or this one. There's values are quite high, or maybe this one. Manhattan plot like this suggests that there may be a quality control. Problems with individual variants that are not supported by their neighbors, especially in case of control analysis, where cases and controls are genotyped separately. Strict QC must be iterated until Manhattan plots looks clean. So it must be clean, only that otherwise there will be so many dots, so many associations will be there, but we have to take care of only those associations which are giving us the clear-cut message, or which are having the good association with the trait of concern. There's a common softwares package for association mapping like Tessel, Gepit, SAS, R, Structure, Spark, or P-Link. There are a number of softwares that are available, and you can use them. And they have the certain advantages and disadvantages of each software. But like you can see this based on these comments, you can understand that like Tessel, this is free. LD statistics is there, sequence analysis, association mapping you can do, and for that you have not to pay. Like Gepit is also free. Some softwares are not free, so you have to see your limitation also. You have to see whether it is free or it's paid. If it is, so you can go for the different software, different packages. We apply sometimes the different software just to see the association, just to confirm the association, whether that association is existing or not. So Gepit means, what is Gepit? Gepit is Genome Association and Prediction Integrated Tools. This is a statistical package that is run in our software environment, developed by Alex Lipka and Zhu Zhang. Later it was Alexander Lipka that further improved it, and uses statistical tools implemented in other programs like Tessel. Although there are programs that are implement the same statistical methods, size of the data set can be a problem. So if size is big, then we use the different softwares. Gapettes can handle large data sets, file splitted. By splitting the file, you can use the different softwares. Some of these programs have the complex interfaces. Most of you are already familiar with R. It does not require that much code to run the analysis, do all data manipulations and analysis in one common environment. Supporting informations is good. Excellent user manual is there, so you can even do the Gapit with the help of that manual. It's easy to use. Developers are extremely helpful, so you can also take the help of them, and is faster to perform. So when we do this Gapit, so we use sometimes the different models like GLM or MLM, CMLM or ECMLM, super fast CPU. One minute. Hello. अरे मैं भी क्लास कौन बोले? अरे रुको यार, मैं वो दे रहा हूँ. So super and farm CPU and billing. And since when we have the data from the different sources, like if it is from the different environments, from different locations, or if it is from multi-year data, if you have, then to deal with that data, we take the help of this blub, like best linear unbiased prediction models, or we use the BLUE in Gapit. This is what we do. And after the confirmation, when we do this multi-model confirmation by using different models, after the identification of those QTLs or those associations, what we do is we validate those associations with the help of cast markers, or maybe if some other approaches are there before their use in marker assisted selection. So this CMLM is an existing single locus genome scan method for GWAS. CMLM decreases the effective sample size by clustering individuals into groups and eliminate the needs to recompute variance components. Second is GLM, is a general linear model, is another extreme case of CLM where all individuals are considered as one group. It can be simply performed by setting the number of groups equal to 1, like group from 1 is equal to group to 1. Means only one group will be there, all will be considered as one group. Third is like mixed linear model, MLM. MLM includes both fixed and random effects, including individuals as random effects, gives as an MLM and ability to incorporate information about relationship among individuals. When you will perform this, I think you will learn the practical classes about the different models which we use in case of Gapit. Like mixed linear model, it includes both fixed and random effects. So there are different things when we, which we use in case of Gapit. And there is super. Super uses the bin approach to select associated markers. The entire genome is divided into equal size bins, and each bin is represented by the most significant markers on the bins. Fast LLM is the newly developed algorithm in GWAS that can solve the computational problems, but requires that the number of SNPs be less than the number of individuals. To overcome this shortcoming, Super extracts a small subset of SNPs and use them in the fast LLM. This Super not only retains the computational advantage of fast LMM, but also rememberably increase statistical powers. Another which is very famous is farm CPU. This is false positive control and confounding between the testing marker and cofactor simultaneously. It can solve the problem of that. And a iterative method named fixed and random model circulating probability uniformation farm CPU was developed in 2016. The associated markers detected from the iteration are fitted as the cofactors to control false positive for testing the rest markers in the fixed effect models. There are many things, many analysis which performs in the backgrounds, which we cannot know, only computer person can learn about. Maybe they are quite familiar with that. They learn all those things. Only thing is, the good thing with this different models, especially with farm CPU, is it takes care of false positives. So would be false positive would be very, very less, or maybe you will not find any false positive there. Next model is multi-locus mixed linear model, MLMM. Gapit implemented MLMM, which use forward-backward stepwise linear mixed model regression to include associated markers as covariates. Another model is BLINK. BLINK method was designed to have both high statistical power and computational efficiency. It was inspired by farm CPU method. It's an advanced version of farm CPU, you can say, with two major changes to achieve the objectives. One is to eliminate the assumption that causal genes are evenly distributed across the genome, which you will not find. So you can, I think that can be solved. That problem can be resolved here. That requires by farm CPU. And the change is to use Bayesian information content. This Bayesian information content, BIC, of a fixed effect model to approximate the maximum likelihood of a random effect model to select the associated markers among the markers remained the exclusion based on LD. As both the models of the testing markers and the selected association markers as cofactors and are fixed effect models, the computation complexity reached the maximum. So, with the multiple models of the implemented in Gapit, a common question is which to choose, which marker system we should select. Internal computing efficiency and statistical power should be the criteria for the selection. Two models use the fixed effect models only, which is the most computing efficient, including GLM and BLINK. Farm CPU is a hybrid type of model that use both the fixed effect model and the random effect model. The rest use a fixed and random effect models, which is computationally expensive, including MLM, CLM, Super, MLM, and CMLM. So regarding statistical powers, multiple loci models like MLMM, farm CPU, or BLINKs are superior to the rest. Within multiple loci model category, farm CPU is superior to MLMM and BLINK. So these are some of the success stories where this has been used. Some of them where we have implemented this here also, and in the outside of the country, where people have utilized these germplasm lines, the genetic resources conserved in the gene banks across the globe, and they have identified the QTLs, and even they have validated. If we see this example first paper, we have even validated those QTLs which we identified in a set of 400 lines, 400 germplasm lines. Most of them were of Indian origin. And we also performed this second study for in the leaf rust, where we also identified the QTLs for leaf rust resistance. This paper we published in Heredity. We also identified, there are also some other studies like Chorosia, here with Dr. Amit. She identified this QTLs for salinity tolerance. And also this Dr. Jyoti, actually one of her students performed this for this root architectural traits, identified the QTLs for that. And there are some other studies where we have identified QTLs like for root laser nematode or for agronomical traits. So there are a number of studies which we have performed here in India, as well as the studies which have been performed outside of country, where people have utilized the germplasm lines or the genetic resources to identify the QTLs. In addition to that, they have also identified the lines where number of favorable alleles were there. So these these lines can be used directly in the breeding programs where we are getting the most of the favorable alleles. So Jiva's promising many disease traits are that we traits are influenced by the genetic factors. They are caused by sequence variants in the genome. Over 12 million SNPs are known in the genome. So question is how to make the selection. I think we've already learned about that. Cost of genotyping is reducing as far as NGS platforms are concerned. So I think it's not quite difficult now. Earlier it was really difficult to perform SNP based as a this genotyping and this GWAS studies. A large number of cases and controls are now available. There is statistical power to detect the variants with modest effects. So we are now the tools also to identify the false positives. We also tools to perform this kind of studies. There are certain issues in GWAS like population stratification. So this we have to take care of whether our population, if there's some outliers are there, so maybe we have to remove those outliers first before performing GWAS studies. And if it is a value which we have taken from the different locations, or from its a value, the phenotypic value we have taken from the different years, I think we have to first normalize the values, applying different tools. And we have to take care of gene to interaction, which might be quite high. And because of that, we can have the lower heritability values. And cost is, of course, a great concern. These are some of the successful stories that I told you that we have performed this GWAS here in India, here in NVPGR, where we have performed, we identified reliable QTLs after having studies for continuously for years. We have evaluated these lines in the field, and then we have performed this by using different models like multi-model analysis we have used, so that the QTLs which we have identified, we can say these QTLs are really the consistent QTLs, which are there using, here if you see this, for example, for spot blotch, we use the TESEL and GAPIT GWAS models to identify the reliable marker trait associations, which we identified. We have even validated them through CAS markers. This study we have published in Heredity Journal, where we have identified these QTLs through GWAS for leaf rust assistance. This is for drought tolerance. This is for S-type rust assistance, again in Indian lines, including land races. This was for terminal heat tolerance for different economic traits under extremely extreme conditions. And this paper we published in Frontiers in Plant Science. So in conclusion, what we feel that NVPGR gene bank consists of diverse set of collection germplasm lines, which are to be evaluated, characterized for different economical traits. Chance of getting novel QTLs or the germplasm lines is quite high. And that will further strengthen our breeding programs, national breeding programs, including major cereal crops. And the molecular markers which we have already validated can be used in marker assisted selection to make the selection more easy and effective. Thank you.
Thank you, sir. And if there are any questions, please post them in the chat box, or you may also directly ask to sir. Sir, so we have two questions in the chat box currently. One is that Dr. Vinod Kumar, he is asking, kindly suggest the book for molecular breeding.
A very difficult question. I have not identified any book where I can get all these, this the content. Maybe this thing you can identify. Maybe in Google is the best guru. Google Guru is the best guru. You can find all such informations there. And whenever you will go in the these programs, you can find even the helpline through their help. You can find the answer of these all things.
Okay, so next question is in perennial fruit crops, no inbred available, where at most of the loci are heterozygous, including the loci for major traits. In such cases, how to deal with heterozygosity? What should be minimum maximum threshold, or any model is there to deal in such situation?
Uh, well, I'm not dealing with the perennial crops here. I'm dealing with the wheat. So I'm not familiar about the nature of that particular crop, perennial crops. But what I can suggest is, since there's too much heterozygosity, first they have to take care of this heterogeneity as well as this heterozygosity. Heterogeneity first they have to make it. If it is only heterozygous, then I think they have to first develop the lines which are homozygous in nature, or there are some programs, like as I told you, which deals with the heterozygosity. So maybe you can split this genome in a small parts, and then I think it can be dealt with that heterozygosity. Okay, minimum and maximum threshold values. Well, dear, there is no minimum and maximum actually that you have to decide what with the value you will consider the this minimum threshold.
Okay, so
Mm.
any other questions? You can type and drop in the chat box. No questions? Okay, sir. So now no more questions are coming. So I think they understood everything, or they have not at all listened.
Some things are very complicated. Even I am not. I don't know even the what is there behind that statistical
Yes.
equations.
Yeah, some of the things we just know the interpretation, no? Only what is being done statistically, we don't know actually, no? What software is doing.
Yeah.
Yeah. Okay, so now we are receiving thanks from the participants. Okay, sir. So with this, I would like to thank you for presenting your talk. And you really gave a detailed account of genome-wide association studies, different platforms used, and statistical softwares that can be used. So I hope that all the participants have enjoyed. Those, at least those who listened to you. And with this, I really want to thank you for being with us and providing your valuable time. Thank you so much, sir.
Thank you, Mamta. Thank you very much for giving me this opportunity to say something about Jivas before participants. It was really, I think it's a kind of honor for me
Thank
to
you, sir.
express my views on Jivas. Thank you very much.
Okay. Okay, participants. So we have our next session to start by 11. So we will be back by 11.
Should I leave now?
Yes, sir, sure.
Okay, thank you.
Thank you, sir.
Thank you.
Please be ready for the next session and be on time. This is the request to all the participants. Thank you.
This 21 days. We welcome you in this 21 days training program. I really feel privileged to get opportunity to introduce our speaker, Dr. H.K. Dixit. Just I am sharing the slides of introduction. Is it visible? Okay.
Yes, sir. Yes.
Okay. Dr. H.K. Dixit is presently serving as Principal Scientist, Division of Genetics, ICR Indian Agricultural Research Institute, New Delhi. His primary research interests includes genetics and breeding of pulses, development of varieties of lentil, mung bean, and rajmas, biofortification, phosphorus uptake and utilization efficiency, abiotic and biotic stresses breeding, and nutritional improvement. The major achievements what he has gained, he has significantly contributed to the development of 15 varieties of lentil, mung bean, and rajma. He has registered total 12 genetic stocks with the PGRC, ICR. He has published more than 200 research publications, including technical bulletins, research papers, review papers, and all. Besides this, he has also edited 3 books. He received many awards like Dr. Rajendra Prasad Award in 2020 of ICR, A.B. Joshi Memorial Award in 2022 by Indian Society of Genetics and Plant Breeding, Dr. Ramnath Singh Award of IRI. He also is a recipient of ICARDA Special Recognition for Research in Lentil. He is also in the editorial board of Indian Journal of Genetics and Plant Breeding and Journal of Food Legumes. He also received a fellowship of National Academy of Agriculture Science, Indian Society of Genetics and Plant Breeding, and Indian Society of Pulse Research and Development. Besides this, he is actively involved in the teaching and guiding the postgraduate student of ICR IRI. In fact, he is also my teacher. So I have also learned many things from him. With this, sir, I would like to welcome in this 21 days training program, and I request you to share your presentation. Thank you.
Thank you, Dr. Kuldeep. So the topic for today's lecture is hidden hunger. We all know that by now what hidden hunger is. It is the lack of micronutrients like iron, zinc, iodine, vitamin A, and folates in diet. And when these micronutrients are lacking in the diet, that impairs mental and physical development, especially of children, adolescents, and females. And it can cause anemia, lowering of IQ, reduced productivity, increased risk of illness, stunting, blindness, and goiter. Goiter is caused due to deficiency of iodine. Globally, 2 billion people suffer from iron and zinc deficiency, and more than 7 million children under age of 5 annually die directly or indirectly due to this malnutrition. When we consider the level of malnutrition at global level, we find that mostly Asia, it affects more in Asia, Africa, and parts of Latin America. The situation is very, very serious in some parts of Africa. The main reason for this hidden hunger is low income. People with low income, they spend most of their income on procurement of staple food, and they rely heavily on staple food for their energy requirements. And staple food like wheat, rice, etc., they are low in micronutrients. So we can see that the people who cannot afford this diversified diets comprising cereals, pulses, milk, animal meat, etc., they suffer most from this hidden hunger. And more than 3 billion people suffer around the world from this hidden hunger. And hidden hunger weakens immune system, leaving people vulnerable to infections like diarrhea. And hidden hunger can also cause stunting, blindness, anemia, poor pregnancy outcomes, and developmental impairments in the children. Most affected from this hidden hunger are school children and women. Uh, when we see that this hidden hunger not only affects the individual, it affects the countries also. For example, in India, hidden hunger causes annual loss to the GDP of about 12 billion. Similarly, in Pakistan, the annual loss to GDP is around 3 billions, and in Bangladesh, 700 millions. And the situation is critical in Rwanda, Tanzania, Zambia, Democratic Republic of Congo, and Nigeria. Harvest Plus has estimated that if we invest $1 in nutrition program, then we get benefit worth $17. So it is worthy to invest in this nutrition program. This will not only improve the public health, but losses to GDP can also be minimized. Then in 2015, United Nations, they proposed 17 goals for developmental agenda till 2030. The basic objective of these developmental goals was to eliminate poverty, hunger, and malnutrition. 12 out of 17 goal indicators are related to the nutrition. Goal 2 is specifically addressing hunger and nutrition. So at a global level in United Nations, this consensus was developed, and 17 sustainable developmental goals were proposed with the objective of removing poverty, hunger, and malnutrition. And significant progress has been made in achieving these goals. However, it is very difficult to achieve all goals by 2030. And then coming to different micronutrients and what role they have in human body, and their deficiency results in what, that I have tried to summarize in 2 slides. Iron is required for oxygen transport and cellular respiration. It's found in hemoglobin, myoglobin, enzymes, and cytochromes. And iron is necessary for cognitive function and optimal growth. Iron status of human being can be measured by hemoglobin concentration and ferritin concentration. Hemoglobin concentration generally is used to determine whether somebody is anemic or not, and ferritin reflects this body iron store. That means iron is stored in human body. We don't have to take it on a regular basis. Iron deficiency can cause this anemia, disrupting endocrine and immune system. During pregnancy, fetal growth and development is affected due to iron deficiency. Maternal iron deficiency may result in premature birth and lower body weight. Second most important micronutrient is zinc. Zinc is necessary for immune system function, protein and DNA synthesis, cell division, and activity of nearly 200 enzymes. There is no consensus on how we can estimate the zinc, but generally stunting of this children, stunting and wasting of the children is used as indirect indicator. As early as 1961, zinc, it was identified that zinc deficiency in human can cause adolescent nutritional dwarfism. A reduced zinc intake can cause mortality due to diarrhea, malaria, and respiratory infection. So even during COVID-19 period, these doctors, they were suggesting intake of zinc. Then coming to vitamin A, in vitamin A, there are 2 forms. One form from animal source form is retinol or retinyl esters, and plant source is provitamin A carotenoids. Provitamin A carotenoids are converted to retinol or retinoic acid in human body. Vitamin A is necessary for vision, immune function, reproduction, epithelial integrity, cell differentiation, and organ and bone formation. Vitamin A deficiency can be measured by studying serum retinol. Vitamin A deficiency can cause xerophthalmia and increase mortality in children in developing world. And it deficiency also increases vulnerability to diarrhea, measles, and respiratory infection. Iodine fortification is very common in India in salt. We get Tata salt, which is fortified with iron. Iodine is mainly stored in thyroid gland, and it synthesizes thyroid hormone, which regulates human growth and development. We require, if we take less than 10 to 20 microgram of iodine, then it can cause hyperthyroidism. Iodine status is assessed from urinary iodine content. Pregnant females and infants are at highest risk of iodine. Fetal growth and postnatal central nervous system growth require thyroid. Then in this series, last is folates. Folates are natural and synthetic. Synthetic folates are folic acid, which is more bioavailable than natural folates. Folates are required for synthesis of purines and thymidinates, which are involved in stability and repair of DNA synthesis. Folates are assessed by folate concentration is plasma, serum, and erythrocytes. During pregnancy, folate deficiency can cause this anemia, lowering birth rate, and preterm delivery and fetal growth retardation. Now coming to the legumes, we know what is biofortification and what are the micronutrients and what deficiency they can cause, and deficiency which results in disease due to low intake of the micronutrients. So coming to the pulses, they help in eliminating hunger and nutrition of human beings, and at the same time, they help in enhancing ecosystem resilience and ensuring environmental sustainability. Pulses, 165 million children are globally are stunted and 101 million are underweight. And malnutrition related deaths are nearly 20 million worldwide. So pulses can help a lot in reducing these MNDs, as they are rich in protein, dietary fibers, complex carbohydrate, resistant starch, and folate, and then micro and macronutrients. Pulses can enhance ecosystem resilience and ensure environmental sustainability, as they contribute to soil health. When we grow pulses, they fix this atmospheric nitrogen, and at the same time, leaves of pulses fall on the ground, increasing the carbon content of the soil. They help in mitigating climate change and improve productivity. And in rotation, pulses reduce fertilizer consumption and decrease greenhouse gas emission. When we grow rice or wheat, we have to spray the urea. Urea is responsible for emission of nitrous oxide in atmosphere, which is very, very dangerous for this climate change. Total pulse production in India during 2021 and most recent pulse production in India was 27.75 million tons. So in pulse production, we have witnessed a pulse revolution in last 10 years. In last 10 years, pulse production has increased by almost 16 million tons, by 11 million tons. When we compare the nutritional value of legumes and cereals, we see that for micronutrients, this chickpea, cowpea, lentil, lupins, mung beans, up to soybean, we can see peak is higher as compared to cereals. Similarly, in case of macronutrients, pulses are better than these cereals. And when we compare folates of these legumes with that of cereals, we find that legumes are rich in folates. Similarly, when we compare essential amino acids of this,
Sir, excuse me, sir.
huh?
Sir, slide is not moving.
Slide is not moving.
Yes, sir. It is still at lack of critical macronutrients in the diet. Only this slide is visible. Other slide is not visible.
Okay, then I will share again. Huh?
Okay, sir. Okay.
Now is it moving?
Yes, sir. It is moving.
So should I repeat again or what to do now?
No, sir. Just one by one brief में कर दें सर. थोड़ा थोड़ा बता दें.
अच्छा ठीक है. हाँ?
हाँ, जो पहले हो गई सर उसको थोड़ा थोड़ा जल्दी कवर कर दी.
ठीक है.
जी सर.
In second slide, I'm trying to tell how this MND, how they are impacting what is their magnitude at global level. When we see that most affected is Asia and Africa and parts of Latin America. In these three continents, it affects almost 3 billion people. And hidden hunger weakens the immune system also. But when it weakens the immune system, then we are vulnerable to infections like diarrhea, etc. Diarrhea, malaria, and even we saw during COVID times also, doctors, they were suggesting this diet of this diet and medicines of for zinc. So we see that hidden hunger can lead to stunting, blindness, anemia, poor pregnancy outcomes, and developmental impairments in children. Most affected from these MNDs are school children and women. Slide is moving.
Yes, sir. Slide is moving.
Okay, now it's fine. Huh? Estimated loss to GDP from hidden hunger has been estimated by Harvest Plus in Asia and Africa. When we see this loss in India, the loss to GDP is 12 billions, whereas in Pakistan, the loss to GDP is 3 billions. In Bangladesh, it is 700 millions. Similarly, in Africa, the loss has been estimated for Rwanda, Tanzania, Zambia, Democratic Republic of Congo, and Nigeria. In 2015, United Nations, they suggested 17 sustainable developmental goals as global agenda for 2030. The basic idea of this sustainable goal was to eliminate extreme poverty, hunger, and malnutrition. When we see this 17 sustainable developmental goals, we find that goal 2 specifically addresses hunger and nutrition. And in 195 countries have committed in United Nations to work on these sustainable goals and to improve this poverty, hunger, and malnutrition status. Then I discussed on what is the role of iron for human beings. Iron is required for oxygen transport and cellular respiration, and it is necessary for cognitive function and optimal growth. Iron status in human beings can be assessed by hemoglobin concentration and ferritin concentration. Hemoglobin concentration is used to determine this anemia, and ferritin is reflection of body iron store. Iron deficiency can cause anemia, and if it affects the fetal growth and development, and maternal iron deficiency can lead to premature birth and lower birth weight. Zinc is very important for immune system function, protein and DNA synthesis, cell division, and activity of nearly 200 enzymes. For measurement of zinc, there is no consensus among the scientists, and stunting is regarded as indirect indicator of zinc deficiency. As early as 1961, a zinc deficiency in human was recognized to be the reason for adolescent nutritional dwarfism. Zinc deficiency can cause diarrhea, malaria, and respiratory infections. Similarly, vitamin A deficiency also can cause xerophthalmia, increased mortality in childhood in developing world, and vulnerability to diarrhea, measles, and respiratory infections. This vitamin A occurs in two forms: from animal source, it is in form of retinol or retinyl ester, whereas from plant, the source is provitamin A carotenoids. Provitamin A carotenoids are converted into this retinol or retinoic acid after utilization by human body. Vitamin A is necessary for vision, immune function, reproduction, epithelial integrity, cell differentiation, organ and bone formation. Iodine is stored in thyroid gland and synthesizes thyroid hormone, which regulates hormone growth and development. This iodine status of iodine can be measured by urinary iodine content, and pregnant females and infants are high risk of iodine. Fetal growth and postnatal nervous system requires thyroid hormone. And then coming to folates, folates are natural, which are found in plants, and synthetic, which are synthesized in the factories. They are folic acids. Folic acids are more bioavailable than folates, which are found in plant. Folates are required for synthesis of purines and thymine dilates, and are involved in stability, repair, and synthesis of DNA. Folate concentration is assessed in plasma, serum, and erythrocytes. And folates deficiency during pregnancy can lead to megaloblastic or macrocystic anemia, lower birth weight, preterm delivery, and fetal growth retardation. Slides are moving. Then
Yes, sir. Yes, sir.
ठीक
Yes, sir.
है. Then coming to the pulses,
Yes.
they have two roles. First role is they help eliminating hunger and malnutrition. Why hunger and malnutrition is important? Because 165 million children are stunted globally and 101 million are underweight. And malnutrition, which is diet related, causes about 2 million deaths worldwide. Pulses can really check this hunger and malnutrition, as they are rich source of protein, dietary fibers, complex carbohydrate, resistant starches, folates, and macro and micronutrients. Pulses enhance ecosystem resilience and ensure environmental sustainability, as they contribute to healthy soils, climate change mitigation, and improve productivity. And in rotation, we see that pulses reduce fertilizer consumption, because nitrogen is fixed by pulses through rhizobium, and they decrease greenhouse gas emission also. When we grow pulses, we apply less urea, and then that reduces the emission of nitrous oxide in the environment. Total pulse production in India last year was 27.75 million tons. In last 10 years, production has increased by 11 million tons and country has witnessed pulse revolution. Now, when we compare the micronutrient, macronutrient, and folates of among grain, legume, and cereals, we see that micronutrients are higher in legumes as compared to the cereals, and similarly, macronutrients are also higher in legumes in comparison to cereal, and folates especially are very, very high in cereals, in legumes in comparison to the cereals. Micronutrient, then essential amino acid profile, when we look at essentially amino acid profile of pulses is also better than cereals. So we can say that pulses, they are much better food, not only for human beings, but also for the environment. To eradicate or to reduce micronutrient deficiencies, we have 4 options. The first and best option is dietary diversification, but all cannot afford this dietary diversification because then we have to eat diet which is rich in fruits, vegetables, cereals, and this meat, milk, etc. Because nearly 10% of people in the world earn less than $2.15 per day. So with very, very low income, it is not feasible to have dietary diversification. So the other options available are, first is biofortification in the field, in the crop or the field. This means that we can biofortify our crop either by breeding or through genetic modification or by spray of these micronutrients on the standing crop. And second option is fortification. This is done in the factory, and at the time of food processing, micronutrients are added. The best example of fortification is salt. Tata salt, which we eat, is fortified with iron fortified. And then fourth option is supplementation. It is on plate. It's called on plate because this dietary supplements, vitamin B, C, iron tablets are given to the people who need it most. Initial intervention to deal with the micronutrient deficiency was that distribution of iodized salt in endemic areas and administration of dose of vitamin A to young children, and distribution of iron folic acid tablets to the vulnerable groups. These vitamins and iron folic acid tablets, they were distributed in primary health centers in India across the country, and now we have only iodized salt. Other salt is not being sold in the market, but these did not make much impact. These intervention could not make much impact because this fund allocation for these activities were inadequate, and in fact that was nearly 10% of actual needed, and allocation of supplies were short of requirement due to poor orientation or training. And the functionaries were not adhering to the guidelines. So that led to total failure of the program. And I cannot say it was failure, but it could not succeed. It succeeded partially. And then there was inadequate monitoring. No proper monitoring was done. And that led to the fact that midterm course correction could not be made. Then Government of India proposed National Nutrition Policy. In this policy, focused approach was adopted. it was a mix of short-term supplementation and food-based strategies, and people were encouraged to grow vegetables and fruits in their kitchen garden, and at the same time food fortification was started. Wheat flour was fortified, and then salt was fortified, and then innovative approaches in information, education, and communication for making intervention sustainable were adopted. So now coming to how this biofortification is done for the food crops. It's just like any other breeding program. First, we identify we identify micronutrient deficiency in a region. These micronutrient deficiencies, they are not uniform across the country. In some regions, you'll find they are more prevalent, and in other regions, they are less prevalent. So first we identify what is the micronutrient deficiency, then how we can address that micronutrient deficiency, which crop we take for biofortification, and what is the baseline. Baseline means the level of that micronutrient concentration in available varieties, or the varieties which are being grown. Then we screen germplasm, and we try to identify the donor which has high concentration of the target micronutrient. Then we also check phytoavailability of micronutrient in the soil. We do pre-breeding if that micronutrient is available in this non-adapted background. So to bring the genes regulating micronutrients from non-adapted background to adapted background, we do pre-breeding. Pre-breeding increases the breeding cycle. And then we do gene discovery using biparental association mapping or genotyping by sequence, any approach. Then after we use the genes which we identify for this micronutrient concentration, that we try to bring in the desired agronomic background through conventional breeding and coupled with genomic assisted breeding. Transgenics for this biofortification, they are not permitted in the country. Country had started this allowing transgenics for commercial cultivation on case-by-case basis. Recently, they have allowed it for mustard variety DMH11, mustard hybrid DMH11. Then once the products are developed, then we do this multi-location evaluation. And if the product outyields the chick variety by 10% or more, or that varies from crop to crop, then the variety is released and seed is multiplied and disseminated to the target population. So Harvest Plus has initiative in 8 different crops. Harvest Plus has this biofortification initiative in 8 different crops. In beans for iron and zinc. Iron was a primary micronutrient, zinc was secondary. In Rwanda, Democratic Republic of Congo and Uganda. Similarly for lentil, iron and zinc in India, Bangladesh, Nepal. For cowpea also, India, Bangladesh, Nepal. Primary micronutrient was iron and then zinc. And this initiative was there in 5 more crops. So when the breeding program started, first in India on cowpea, the baseline which was estimated was around 30 ppm, and target increment was suggested of plus 33 ppm. So target level in crop was 63 ppm. This target increment we set on the basis of diversity available in the germplasm. Here we consider both cultivated and wild germplasm. Similarly, in case of lentil, this micronutrient was iron and secondary was zinc. And baseline was found to be 40 ppm based on the evaluation of cultivars grown in these countries, and based on germplasm or based on variability available, it was decided that target increment of 30 ppm can be made successfully. And so target level of the crop was set at 70 ppm. Similarly, in case of common beans, this baseline was 50 ppm and target level in crop was 94 ppm. Then in this slide, I try to compile the information available on range of variation for grain iron and zinc and selenium concentration in common beans, chickpea, mung bean, and lentil. Here, the interesting thing is that there are hardly 2 reports in which large germplasm accessions or samples were evaluated. These are in common beans, core collection, and Portuguese accessions. In rest of the cases, limited number of genotypes have been evaluated, and based on that, this range for iron, zinc, and selenium has been published. So unless we evaluate large number of genotypes, not only cultivated, but wild species also, then we cannot improve our this micronutrients in the food in these legumes. So evaluation of large number of genotypes is the basic requirement. Then we see that in chickpea, lentil, common bean, pea, and mung bean, this QTLs, SNPs, and SSR, FLPs, etc. has been reported by different workers. But till date, no marker-assisted product is developed. At IRI, we are trying to develop lentil varieties through marker-assisted selection. Now coming to pulse biofortified varieties. Pulse lentil biofortified varieties have been released in Syria, Nepal, Bangladesh, and India, and varieties are in testing stage in Lebanon, Ethiopia, Morocco, and Pakistan. Actually, this is the program of ICADA. ICADA biofortification program was funded by Harvest Plus for 10 years. During the last 4 or 5 years, they have stopped funding. Harvest Plus, they gave initial start to this biofortification program through CG centers, which are active in country of their mandate region. In India, though we don't have this biofortification program through ICADA, but we have direct funding from Harvest Plus for 4 years of rupees 1 crore. IRI is working on biofortification of lentils for Bihar. For Eastern India, Harvest Plus has received money from Bill and Melinda Gates Foundation. Beans have been developed for large number of countries in Africa and Latin America, and adding to this list are Haiti, South Sudan, Ethiopia, and Malawi. Cowpea varieties are focused. Development of cowpea varieties which are biofortified is being done in India. Dr. B.B. Singh is there, and Pantanagar. Earlier, he was working at ETA. Then coming to dry beans, there is about 79 varieties of this dry beans have been developed in different countries, and you can see there are varieties in Rwanda which have 119 ppm iron concentration, and other variety RWCB 184, it has iron concentration of 113 ppm. So And similarly, in cowpea, there is one variety in Zimbabwe, CBC 60, and in Brazil, 3 varieties are reported. But in for cowpea, that target level of breeding was above 90, but these are having low. But anyway, Harvest Plus presents them as biofortified varieties. So I have shown here. In India, we have Panthalobia 1, 2, 3, 4, 5, and 7. In case of lentil, we have 2 biofortified varieties from Syria, that is Idlib 2 and Idlib 3. From India, we have one variety, L4717 and IPL 220. L4717 is a first variety of lentil to mature in 100 days. It was released from IRI New Delhi a few years back, and now it's leading variety of the country. From Bangladesh, we have Bari Masood series, which are biofortified, and in Nepal also they have recently released 2 biofortified varieties. Then it's not the concentration of micronutrient in the food which is important. Important is bioavailability. If bioavailability of iron in mixed diet is 14 to 18%, whereas in vegetarian diet is 5 to 12%, and in food product is 1 to 40%. So for zinc, it is again 40 to 60% and 20 to 30% in food low in zinc, food high in zinc. So I mean, it's not the concentration which is important. Bioavailability is also important. If breeders, if they cannot increase the concentration, they can increase the bioavailability by reducing the phytic acid and other anti-nutritional factors. So in case of this dry beans or beans rich in iron, we call them iron beans, health evidence have been generated. In Rwanda, these iron depleted females, university students of age group 18 to 27, they were fed with this iron beans for 4.5 months. And after feeding them for 4 or 5 months, it was found that their iron status improved, which was evident from hemoglobin, serum ferritin, and total body iron. And in the same study, it was observed that their memory, attention, ability to do physical tasks improved, and their likelihood of reaching their potential at work, school, and home. In second study, which was conducted in Mexico, among the school children aged from 5 to 12 years, these were fed with iron beans for 6 months, and their iron status improved. So when we see this cultivation of biofortified crops, at the moment, biofortified crops are being cultivated in 12.7 are being cultivated by 12.78 million farmers around the world, and it is proposed that by 2030, nearly 1 billion small holder farming households will grow these biofortified crops. For biofortification, the requirements are we have to increase our breeding effectiveness. Then for increasing breeding effectiveness, we have to increase the scale of breeding. We have to make large number of crosses, and that population sizes should also be very large. And then we have to intensify testing. We can do multi-location testing, and for selected crosses, we can do rapid generation advance, for especially for if we want to do this genetic studies, and then we can grow we can use control environments, and we have to breed in target zones. We have to test our material in target zones. Then we can use modern technologies for trait diagnostics, high throughput screening. For determination of iron and zinc concentration, we can use XRF, that is non-destructive method, and we can use molecular markers and genomic selection for precision in breeding. And we need to have a knowledge on genetics and crop physiology, and national agriculture research and education system capacity should be there for breeding system and extension. Then coming to challenges to the biofortification of food crops, the first challenge is that we have to increase the awareness. We have to increase this awareness of national and international public officials, because this program requires funding, and for funding we have to increase awareness of government officials. Then we have to explain health benefits of biofortified crops to rural and urban population, and we can use these biofortified crops for development of this value-added products, and demand has also to be generated, because farmers, when they go to the market, when they sell their products, there is no premium on biofortified crop varieties. So farmers basically they are focused on the seed yield. Demand for biofortified crops can be generated if these crops are being distributed through PDS system in the most vulnerable areas, and this biofortified legumes or wheats or cereals, they can be given in through PDS system, and slightly more MSP can be fixed, so that farmers will also be encouraged to grow these crops. Then there are some policy issues. Public support for adoption and acceptance is required, and then we have to regulate. We need regulation on food standard, consensus on definition of biofortification. And in India, we have national nutrition strategy by the central government. And for increasing the awareness, there is a change in appearance or poor acceptability. Sometimes grains, they do not look nice. So we have to explain people that if suppose the grains, they are having phenols, they are darker and they may not be preferred by consumers. So if we label the packet that these contain so much things, then consumers will be interested in buying those products. Then farmers' acceptance and sensory perspectives are also there. Government support is required. More trials are required for acceptance, and trainers training on production is required. For research support, there should be minimum levels of micronutrients for release of variety. This in India we have done in case of pearl millet, where no variety, synthetic or composite, can be released if it has less than 42 ppm iron and less than 32 ppm zinc. CG centers and national programs should jointly evaluate this available germplasm, including the wild species, and research on bioavailability is also required for strengthening the program, and public-private partnership is required for upscaling of these varieties. Then coming to the way forward, in way forward, what happened? In way forward, for addressing micronutrient deficiencies, it is essentially required to breed for public health. Here, what we mean to say is that all breeding programs should have biofortification as one of the objectives. And CG centers and NARS must recognize minimum level of micronutrients and vitamins in all crops. That we have done in case of pearl millet, but this is required in other crops also. Then other issue is this national and international public officials should understand the importance of biofortification, because this program is needed for ending malnutrition, and every program requires funding, so they have to provide the funding, and the issue of bioavailability should also be addressed. And then CG centers and national center programs that we should address should evaluate all cultivation for micronutrient concentration. Upscaling of biofortified crops will improve this health by reducing M&Ds, and here we can take help of private sectors and NGOs, and we have to ensure remunerative pricing for farmers. Unless we ensure this remunerative prices, then we cannot ensure that farmers will grow it. This can be ensured by distributing these biofortified crops under PDS system, where slightly higher prices, minimum support prices, can be fixed for biofortified crops in comparison to non-biofortified crops. This will encourage farmers to grow the biofortified crops. Thank you. With this, I end my presentation.
Thank you, sir, for giving very comprehensive, comprehensive lecture on this most important issue of hidden hunger and how we are overcoming its strategies. And your efforts in biofortifying pulses are really commendable. So if you, sir, allow, then shall we take some questions, if any?
Sure, we can take few questions.
Dear participants, if you are willing to ask any question which is relevant to this topic, it would be, please ask any question either through chat box or directly you can ask. Any question is there? I don't see anything in chat box. Okay. If no, then sir, I would like to thank you on behalf of my own and our own organizing committee also. And it's really that you have spared time with us. And I hope all participants have been benefited with your words. And if you are getting any question which is relevant to your topic, then we'll be sharing with you for the answer in this thing. So thank you, sir. Thank you for joining.
Thank you.
Thank you, sir. Thank you very much, sir.
Thank you, madam.
So I request Dr. Mamta for further proceeding on the day. Mamta is around.
Yes, sir, I'm here.
Okay, thank you.
Next session is from 12:15, ma'am. Jyoti, ma'am.
Yes, yeah.
Okay.
12:15.
Yeah.
So there will be a short break.
Yeah, short break of 15 minutes.
Okay, if anything is related to participants, then we can share. Madam?
Regarding valedictory session.
Anything related, if you think there is a need of something.
Questions are not coming. This is the issue.
Because at least
Maybe
one or two questions they have to ask.
Become tired maybe after
Not
listening
tired, but
from so many days.
whoever is speaking, they know some. No, they have some expectation that if nobody is asking question, it doesn't mean that they are not listening in this thing.
That impression should not be there, no?
Exactly. Anyway.
At least few questions should come. So, ma'am,
we'll get back. Bataiye.
You'll be addressing, na, ma'am. Jyoti, ma'am.
Yes, I will.
Okay.
So for tomorrow, I can. Tomorrow we have being Saturday. So tomorrow lectures are very important because they don't have time on the working day. So that's why we have kept. So the morning session will begin with a lecture from the person is from Kansas State University, Dr. Bara Prasad. So in USA, it will be night time. So everybody should be ready by time. We'll we'll begin our program by sharp 9:30. So all should join by 9:20 or so. And then Joint Director Research, IERI, Dr. C. Biswanathan. He will be talking about phenomics. So in spite of his busy schedule, he agreed to deliver the lecture. So tomorrow his talk is also there. Then another talk is by Dr. Jagdish Rane. So now he has been selected as director. Which institute? In Kazri.
Arid horticulture.
Arid Horticulture Institute. So he has also readily agreed our invitation, although he is quite busy, but he told, okay, so means he will be delivering. So all the lectures are very, very important. So means tomorrow there should be good attendance. Otherwise, our purpose will not be served to deliver the good lectures. And afternoon, one practical session is that is also very important related to pre-breeding embryo rescue techniques in wide hybridization. So that is also one of the important session. So I will request participants, whoever is listening, all are not here right now, but I will put a message in chat box. Okay. And Kuldeep, for a valedictory session, that day also
We have
we
to
can,
maintain the attendance, that's all.
yeah.
Nothing more is expected
With
from
the-
participants.
And for feedback, again, 4-5 feedback means we can,
Feedback also, we can inform prior.
yeah, we can inform priorly. So that day, Dr. Tia Sharma, Deputy Director General Crop Science, he will be the chief guest that day. So we should expect good attendance. Okay, so
Not
that's
should, we
all.
must.
That's all from my side.
ठीक है, तो again we can come back on
12-15 minutes.
After 15 minutes, we'll meet again.
Okay. Namaskar, sir.
Namaste.
Namaste,
Namaste.
sir. Good afternoon, sir.
Yeah, good afternoon. Is it still going on? Previous session?
No, sir. Previous session finished, sir. So we had a short break, sir. 5 minutes
Okay, okay,
break.
okay.
Tha. So now.
Yeah, it's fine. It's fine. Yeah.
So how are you, sir? Fine.
Good, good.
Okay, okay. So we thought that means you'll be traveling for RSC and all, as you told, sir.
Yeah, this month is over. Only now, next month it.
Okay, okay, sir. Okay, okay.
Yeah.
So just we'll start shortly, sir. So before
Yeah,
that, I will
sure.
tell
It's,
about the sir, trainees, sir. So
yeah.
now we have, sir, here more than 80 participants, sir, 80 training. We received more than 100 applications, sir, around 120. But
Yeah.
considering the Zoom slot, we kept around 84 participants, sir.
84
Sir.
participants.
So
Okay.
they are mainly from, sir, that breeding background, only breeding and genetics.
Right,
Most of
right.
them are, sir. Working on different crops, rice,
Yeah.
wheat, pulse, vegetable crops, from tree species also. Then institute-wise, also most of them are, sir, from State Agriculture University.
Right. Okay.
Yeah, 59 are from State Agriculture University.
Okay.
ICAR, we have 9. 2 are
Yeah.
from, sir, CG Institute. From ICARDA, we have 2 participants.
Okay.
Then from, sir, private institute also 6 are there.
Right. Okay.
Then some other universities that, and
Good.
most of them are, sir, assistant professor rank.
Okay.
Yeah, around 50 are, sir, assistant professor rank. 12 are associate professor or senior scientist. 5 are, sir, professor rank and principal scientist, and few are, sir, technical.
Yeah, right,
So,
right. So it's a mixed.
Yeah, mixed groups.
Yeah,
Is a
group.
kind of a mixed group. Some of them are heading also some division in their institute
Good.
among those professors.
Yeah.
So now it is the means next week is the last week. So this is the
Okay. Okay.
So before yeah, so dear participants, so our speaker of the today's lecture has joined here. So first I would like to introduce sir. For that I'm sharing one slide. So I welcome you, sir, for this training program to accept our invitation for delivering the lecture in spite of your busy schedule. So Dr. KB Bhatt, sir, he was at NBPGR as a HOD of Division of Genomic Resources. He joined ICAR in 1987 and served ICAR till November 2017. After that, sir served at NBPGR as a ICAR Emeritus Scientist. Regarding sir's education, sir has obtained his master's degree in plant breeding from PAU Ludhiana and PhD from US Dharwad. So sir has vast specialization, vast working knowledge about plant breeding genomics, and sir is a man of population genetics. And so sir is known very much about population genetics and then plant genetic resources, genetic diversity. He has the great role in cultivar identification using DNA fingerprinting techniques, molecular phylogeny tools, etc. Then sir has enormous contributions to the PGR science. So just to name few here, I'm giving some few achievements here. So sir has identified new sources of tolerance to okra yellow vein mosaic virus and okra innocent leaf curl virus tolerance. He was having great interest in the wild species and pre-breeding program, and he initiated the pre-breeding program in cultivated okra and generated more than 175 MP diploids and backcross progeny involving 5 wild donor species, which has tolerance to okra yellow vein mosaic virus and okra innocent leaf curl virus. Then regarding taxonomy, so sir was master of taxonomy also. So he resolved taxonomic ambiguities in species identities for the genera Vigna, Cucumis, as well as Avolmuscus. Then he has also role in identification and description of new species in Vigna and Avolmuscus. So that is NVPGRensis species. No, I have not mentioned here, but I remember the name. Then as a PI consortium of genomics platforms that has launched crop wild relatives portal on the NVPGR website. Then to his credit, he has several awards and honors for serving PGR globally also. So sir got certificate of recognition as best project for the World Bank funded ICR NAIP project, that was biosystematics of the genera Vigna, Cucumis and Avolmuscus. Then sir has been invited to prepare a manual by IPGRI Rome on molecular tools for analysis of genetic diversity in plants. And sir has been RSC member of various institutes, various CSIR institutes, ICR institutes, and SAUs also. So he has been very busy for as a RSC member and guiding several institutes for the research program formulation. Then he is the great teacher. Sir is the great teacher, and he has been the resource person in various training programs of national as well as international level. So for example, CMD, Delhi University, then IPGRS sponsored international training in IARI and several other institutes. Sir has delivered more than 100 lectures. Then regarding publications, to Sir's credit, more than 300 publications, including research articles, book chapters, technical manuals, etc. And regarding citation, so Sir's citation is very much cited by the researchers across the globe. So 5,417 citations of his work are there. So with this, sir, I will invite you to deliver your talk. So again, I thank you, sir, for inviting our.
Thank
inviting,
you.
sir.
Thank you, Dr. Jyoti Kumari. And just sharing my screen. Is it visible? No. Oh, sorry, it's
No,
the
sir,
wrong one, I think.
some
It's
open
wrong one.
file.
Yeah. Okay.
Yes,
Okay. Okay.
PowerPoint is open, no, sir?
Yeah, it's open. Just
Okay.
1 minute. This is fine, visible? Is it visible?
No, sir, no, sir, it has not been shared now.
Okay, 1 minute. Just 1 minute. Sorry for the.
No, okay, sir, okay.
Let's go 1 minute. Is it visible now?
Sir, it's visible, sir, but
Yeah, 1 minute. Yeah, share screen. I'll just do share screen. Now it's okay.
yes, sir, perfectly.
Yeah,
sir.
the slide is
All well,
moving.
sir.
Is the
Yes, sir,
slide?
slide is also moving, sir.
Okay, thank you.
Okay.
Thank you, Jyoti, for the nice introduction. Just welcome to the participants. It's quite a big gathering, I suppose, all the interested participants. It's good that NBPJR has tried to give a virtual training on management and utilization of plant genetic resources, and also included this lecture on importance of population genetics in PGR management. Actually, in this present world of softwares and program-based programs, I presume that most of us do not really care to look into the basics of population genetics. Sorry to say these words, but that's what we get I mean, idea from going through the publications, etc. So I would be just, even though it's a very mixed type of gathering, I'll be starting with the basics and touch about the various aspects, slightly advanced aspects of population genetics, and the how these parameters or statistics that we calculate in population genetics can be used very effectively in PGR management. Actually, when it comes to PGR management, uh, all activities can be grouped under these 4 different heads. That is acquisition of germplasm, maintenance, its characterization, and then utilization. If you go through the literature as well as the various activities I have listed under each of these 4 heads, you can see, you can visualize that in each case you require some empirical data if you want to go about the whole process in a scientific manner. Just give you one example. There is a good book compiled by Dr. Paroda and Dr. Arora. That's our previous director as well as the in-charge director of NBPGR. He was Dr. Arora was a doyen of plant genetic resources uh, survey, exploration, and collection. Basically a basic taxonomist who did lot of survey, and this is a very good compilation. I am sure in your earlier lectures from Dr. K.C. Ebert and others, they must have mentioned this particular compilation, which is a joint publication from NBPGR and then IPPGR, and it was published in 1991. And if you open the first chapter, that's on acquisition of germplasm, they list very specifically various aspects you should take into consideration when you go for survey and collection programs. And they say clearly how many plants and why so many plants or so many sites, etc., have to be sampled, and you need to go for collections. And what's important for us is these numbers that I have highlighted here. And what is the basis for this? The 330 to 100 individuals per site. That's what they say. And when they cite, it refers to the ecosystems or localities where these individuals or the land races or the wild species occur. And these sites have to be as many as possible. That's what they list out. And why as many? It's a bit vague, but then still it gives you very good idea about the enormity of the work that is to be done. And then what's important is try to capture all alleles with frequency of approximately 5% or higher. Why 5%? Why it's not 1%? To capture 1%, actually that has to do with the logistics of the whole process. The number of plants you have to sample, if it is 5%, goes to maybe 100, 200 plants. That's the reason why this is what has dictated this many individuals per site. They what they have recommended. See, the idea of bringing these points was mainly to highlight the importance of empirical data. And how do you generate this particular data of just, let's say, the frequency of alleles? That is where the population genetics comes in. And then to be specific, certain three different aspects of researchable issues I have where the population genetics has direct relevance. I have just listed out. I will be sharing this set of slides with the organizers, and they will distribute it to you. You can use them. See, this may be related to these issues, may be related to conservation, crop breeding, or related to basic issues of evolution. Even though conservation and evolution have direct relevance to each other, the basic way we go about it is different. So it has been listed out separately. I'm not going to read each of these aspects, but then what's important is we should know when it comes to conservation, it is the augmentation or the collection of germplasm. So we should know what is the way the temporal and the geographical way this diversity in a particular a crop species or a plant is distributed in nature, as well as in the gene bank collections. So whether we have sampled, once you have an idea about what is present in this particular taxa, then we can compare it to a real analysis between what is present in the collections and what is available and what is said to be collected. So, and obviously where this type of variation or diversity is available and where we need to give priorities when it comes to collection as well as for conservation. So using only finite resources, how do we ensure for the future our requirements of diversity? So these questions are very important in germplasm management and conservation, which is inclusive of it. And then how can we quantify current diversity for future reference for studies on genetic erosion? See, the present, even though the various organization dealing with genetic resources claim to do a gap analysis in the genetic diversity held in their collections, that gap analysis is mostly confined to the geographic origin of the accessions. When you say a gap analysis, what we mean is the extent of genetic diversity held or collected and maintained. So that aspect is more important, although the place from where they were collected has large bearing on the diversity or the variation that is being assembled. But this aspect need to be taken into consideration. And then does a pattern of distribution exist so that we can have a guide for our collection activities? So when we say pattern of distribution, I'll come later on with an example, classical example of Dobzhansky in their studies. And then can we ensure that our samples are distinct with not too high amount of duplications or duplicate collections? It's a very dicey word to use, duplicates, in germplasm collection, because in the present era of genomics, maybe you can argue that there is nothing like a duplicate in the natural populations. Each plant, even if it is self-pollinated, might be different. So, but then when it comes to the users, that's the crop breeding programs, certain extent of differences get clubbed together and they can be called as duplicates. So whether it is right or wrong, it's debatable. We can keep on debating. So do these apparently different samples belong to different taxa? So that's where the basic identity analysis also comes in. Okay, coming to the way we go about. See, it's important to define our objectives. So let's say in this beautiful set of mango varieties, you have the varieties collected from the various ecogeographies. So you have the North Indian cultivars and then you have the South Indian cultivars. Why I have distinguished these two is because they differ with reference to the basic aspect of the way the flowering happens, whether it is every year or alternate years or erratic. So usually most of the South Indian types, it's claimed that they are regular bearers, meaning they flower every year, whereas most of the North Indian types are supposed to be erratic, but irregular in bearing. Is there any genetic basis for claim these type of interpretations? So that can be done if you have that type of study, whether there is a basis or not. You can find out if you do a good scientific analysis of it. Now, this is one study we had done where we had done over 1,000 collections of mango from 6 geographical groups, 20 districts, just to see whether what is the basis. Is there any differentiation of varieties genetically existing in among these varieties or cultivars? Now, the way you group them and collect them, that's very important in this type of studies. We'll come to this later on. Okay, that is where the basics of population genetics comes in. See, historically, if you go through the evolution of the subject of population genetics, it has inputs from basic evolutionary biology and then the Mendelian genetics, evolutionary biology of Charles Darwin, that has dictated a lot into the basic principles of population genetics, but it was developed from these foundations by the Civil Wright, then Fisher and others into a topic known as population genetics in the early 30s. And that has given rise to, or it dictates various principles related to conservation genetics, and in combination with the conservation biology and with the inputs from ecology, it has evolved into a topic known as molecular ecology. So these basics, I won't go further into it. Coming to the actual topic of population genetics, you might have, I don't know, the whether, see, nowadays we do use all these parameters of allelic diversity, allele frequency, then the population disequilibrium measures, so on and so forth. But have we really been taught on the basic aspects of this? See, when I studied population genetics, the disequilibrium or linkage disequilibrium, it was just it would just find a very small mention in population genetics. But nowadays that has got evolved into a major branch, which is used a lot in when it comes to identifying the markers or the genes that are linked together and which can be used in crop improvement programs. So the mainly the population genetics, we just to put it in record, it is a study of genetic variation within and among populations and the evolutionary and ecological processes that produce or maintain or even eliminate that particular type of variation. So we have two major terms to be distinguished here. That's the divergence and polymorphism. When we say polymorphism, it is the variation or genetic variation within a species. Divergence refers to variation among the species. So this aspect, please bear it in mind. Okay, the basic principle of population genetics derives its inputs from this particular the theorem or the principle given by Hardy and Weinberg, known as population equilibrium principle. And the assumption that in the equation form takes into consideration the respective genotypic frequencies, let's say in a diploid organism with the biallelic nature, if P and A, capital A and small a are the two types of allelic forms in that particular locus, then which are representing or get converted to, it can give inputs to the frequency of various three genotypes of genotypes that are possible. So the homozygous dominant genotypes P square, homozygous recessive Q square, and the heterozygotes, the 2PQ is the proportion, and the sum total of it under equilibrium conditions totals to 1. But this particular equation holds good, or principle holds good, only if these basic 8 assumptions are met. And these assumptions are that, I'm just reading it so that we understand the pitfall of this particular principle as well as its utility. See, the organism we are addressing should be diploid, then there should be sexual reproduction, and the generations must be non-overlapping, and mating occurs at random without any restriction, and then large population size exists, and there is no migration, no mutation, and no selection. See, in principle, if you really study the natural populations, you will realize that probably you may not come across any population or the group of individuals of a species which meet all these 8 basic requirements or assumptions. Then are we just wasting our time looking at this Hardy-Weinberg population equilibrium principle? No, because it has basically 4 major uses. If we use Hardy-Weinberg principle in our studies, it enables us to compute the gene frequency or genotype frequencies from generation to generation, even under if there is selection operating in that particular group of, or in those generations and the group of individuals. And then it also serves as a null model in tests for natural selection, non-random mating, etc., by comparing observed to expected genotype frequencies. We will discuss about these, what actually it means, each of these observed and expected genotype frequencies and various statistics later on. And then it also helps in tracing the origin of the various individuals, like in the case of forensic analysis. Then the statistics of expected heterozygosity provides a useful means of summarizing the molecular genetic diversity in natural populations. So these things we must bear in mind, that even if the 8 principal assumptions are not met, assuming that we can go ahead with the analysis or applying these principles, assuming that these assumptions are met, and then try to see which of those from the tests of significance each aspect can be tested, and see whether the population, first aspect that we do is to test for existence of equilibrium in the populations. So if they are not existing, they are violated, they are not, the allele frequencies are not maintained from one generation to another generation, it must be due to any of these 8 basic violation of any of these basic 8 assumptions. So you can test for them and see which one is actually relevant, or which of them are relevant in those populations. So that helps us in identifying exactly the reason for deviation from equilibrium principles. And then to see, you can also reduce from the data. Suppose you find that there is rampant selection operating. When we say selection, we are generally referring to the evolutionary processes of selection through environmental, the factors. So it may be in terms of the temperature, moisture, so on and so forth, even fertility. So you can identify certain genes that are related to these violations. So that, because when you do this, the test for equilibrium, it is done locus by locus way. So you can identify which particular locus there is violation of, or the tests are not getting fulfilled, they are violated. So that helps you in identifying the putative genes or the regions that are probably involved or related to this selection process. Okay, so similarly for migration, you can quantify, and then the mutation also you can go ahead with it and see whether at all it has any relevance to the genetic resources activities. Okay, so as I said earlier, suppose there is departure from Hardy-Weinberg equilibrium principle, the first thing that you need to check is the gene diversity, that's the heterozygosity indices. So if there is high gene diversity, that means different genetic sources due to high levels of migration from a diverse population, genetically diverse population, there is high level of migration that has taken place, leading to greater number of allelic diversity and in turn greater heterozygosity. So that is how you can check for migration. And then if there is a greater amount of inbreeding, what will happen is when you calculate the heterozygosities, you can calculate the percentage of heterozygotes in a population from counting the possible heterozygotes in the population and then expressing it as a frequency, and then compare it with the expected heterozygosity. Suppose there is observed heterozygosity is greater than expected heterozygosity, that means there is more outbreeding taken place in comparison to what is expected in terms of population equilibrium principle. So conversely, if there is greater inbreeding, then the observed heterozygosity will be far lesser than the expected heterozygosity. So that's mainly because the number of heterozygotes you see or count in a population or that exists in the population will not be in proportion or relation to the expected heterozygotes that are calculated based on the allelic frequencies. Okay, I don't know. I think I have not included the basic way it is calculated. If you want, we can go to that aspect later on if somebody has any questions regarding that. But there are any number of at least 3 basic good quality programs which can help you in softwares that help you in doing this type of calculations. And then if there is a possibility that there is asexual reproduction that reduces the random mating or the proportion of random mating that happens. So that also you can identify. And then the restricted dispersal in case of lack of gene flow, then you can attribute it to restrictions on the dispersal. There are several other aspects that also you can derive or infer from the data that you have and the statistics that you calculate. Now, in order, if you want to do all those calculations, what's important is to define the populations near correctly. It is absolutely not possible to define a population very effectively with the 100% surety, but essentially because it requires prior study, and then you conduct the tests for the goodness of fit of your hypothesis, and then say designate the population, like in this particular case. Hello, anything? Hello, somebody is asking.
Hello, हाँ sir.
Hello. Yes, anything?
Generally, they ask in the end, but so if dear participant, if you like to intervene in between, so you can ask to sir
Yeah,
in
you
between.
can ask. You
Wherever
can, no problem.
if there is dot, so sir will clear in between.
Probably you wait for some more slides. Some aspects might have got explained in the later slides also. Okay, but then few things, basics I have not included because it becomes too vast.
Yes,
Is there any question or can I go ahead?
sir, you can go ahead.
Okay. Now, defining a population, if you look at this particular valley, let's say this, say why this river valley? Essentially because the natural dispersal and the ecosystem is dictated by the presence of suitable ecosystem for a particular plant. So suppose these are three tributaries of a same river, and the three river tributaries, whether you, and in all these places you have that specific population and the species present existing in nature, and do you call them a single species? Now that can be detected. See, our first impulse is to designate them as three different, so not species, populations. Our basic instinct will be to designate each of them as three different populations. Is it correct? See, a population by definition says that it's a local group of conspecific organisms sharing a common gene pool. All of them might be sharing a common gene pool, but are they isolated reproductively? If they are reproductively isolated in terms of presence of hills and other geographic barriers, so on and so forth, or presence of even forests in between, that also isolates these organisms or the individuals residing in different localities. And in that case, you can designate them as three different populations. In its absence, if it is a plain no barrier present, then it is safer to designate them, all these groups of individuals, as belonging to a single population. So the basic question, you can always do a test. So you can group them as in three different ways. That is, all of them as are single population members representing a single population with, if there is free gene flow or migration of individuals or the pollen or any propagules from one group to the other group. If there is it is completely absent, the gene flow or migration, then safer to designate them as three different populations. Suppose you find that no barrier present between this and this B and C groups, then there is you find that there is a possibility of migration due to whatever reason, animals, birds, or even pollen, then safer to group them as single population and this as a different one. So you have two populations. So these things can be tested. So that is the beauty of the population genetics. I'm not going to how you test it. If you want, I will show you these relevant slides later. Okay. Now, if you are sampling, how do you go about sampling them? In the second slide, I said I referred to Dr. Arora's publication write-up on the 30 to 100 individuals to be sampled from each site. Now, if it is a single population, these are 3 different sites, but do we go for intensive sampling in each of these sites or moderate sampling in each site, so that total we are sampling maybe 100 200 individuals. So if you are sure that there is gene flow happening, it is safer to sample equally and randomly in these ecosystems or the localities, and that will give you a better representation of the population and the better representation in your sample that you are using. Suppose if there is you find that the population size in each of them there is, due to whatever reason, the population size it varies drastically, then maybe you use your discretion and then try to, if the number of individuals is very less in some locality, then maybe you can go for sampling equally through whatever procedure in each of those localities and sample each individual and constitute a sample for your analysis or the further work. Or if the size is too high, then you can go for the basic ways, random or zigzag sampling or so on and so forth, that might have been discussed earlier lectures. So that is how you can go for doing this sampling processes. Okay. Now, this is about sampling within populations. Now, when you do the analysis to draw or come out with empirical data, now the basic question that you always will have is whether you use a morphological characteristics, set of morphological characteristics, or the present day, the molecular or the genomic tools for it. The basic question that comes to your mind is how many loci you should assay, and whether you should go for the randomly distributed markers or any selectable markers related to, let's say, a functional polymorphism that is present, or whether you go for only selectively neutral markers. So these questions can be answered by the basic objective that you have in mind. Like if you are going for, let's say, abiotic stress tolerance, or let's say specifically moisture stress tolerance, you have umpteen number of the genes information that are available to you in that any related or the same crop species that you are working on. And it is safer and will be more specific to work with those markers or those gene loci and the type of variation that might be existing in those loci. So that is the basic way I'm just trying to guide you in this particular aspect. Or if your idea is only conduct a basic genetic diversity analysis, I would prefer to go for selectively neutral markers. See, selectively neutral markers have an advantage in the sense that they conform with the basic evolutionary requirements or the principles, and then you can use it to draw conclusions on the distribution of diversity and the changes that might be happening across the time zones, as well as the ecogeographic, and also related to various other aspects of evolution. So that is why selective markers are better and more informative from this point of view. Okay, what will happen if we just collect, let's say, few individuals, or collect seeds from just 10 individuals or 30 individuals or 100 or 1,000? Actually, this 1 lakh individuals, it is extremely difficult to go for sampling. So let's say, if you compare between these finite numbers, that's the 10, 30 and 100, what happens to the estimates that you derive or the do from compute from using those samples? See, you see that the one type of allele, if in the if the sample size is just 10, it may give you an estimate of 0.75. But when the number increases in the sample that you have used in the analysis, it comes to, let's say, 0.64. So it is more equal frequencies you are coming at or arriving at. So obviously, these differences in the allelic frequencies that you estimated using smaller sample number are errors in the estimations or the bias due to biased sampling. So this is one aspect that you always have to take into consideration. So what exactly is the optimum sample size that should be used when you go for collection, as well as when you do this type of analysis? See, if you have, let's say, comparatively low frequency allele, which we generally consider as rare, they will have a frequency of 0.1. So if you want to capture that particular allelic frequency in near equal quantities as it might be existing in the natural population, then what is the number of individuals you need to collect and analyze? That's what is given in this particular table. It's attributed to Gregorius. It was published in 1980. So there are different tables that he has come out with in that particular publication, and it actually depends on the type of genetic control of each of these markers that you use. This is specifically for a codominant marker system. So in a codominant marker system, if you want very high probability of estimates for the frequencies, then at least 33 individuals need to be collected and analyzed. So when you say 33 individuals to be collected, it is 33 random individuals. It is not 33 seeds collected from a tassel or a panicle or any propagule, a set of propagules from a single plant. So this point must be taken into consideration. And you will see that when you come to the practical analysis of what you have collected, these points that I'm trying to drive home, it has larger bearing. Okay, then coming to the second best example in that particular case is, let's say you have collected a particular wild species sample or the populations you have surveyed, and let's say it is Vigna sublobata, and the idea was to collect the virus, yellow vein mosaic virus tolerance mechanisms in those wild relatives of radiata. And you find that we plant it in hot spots, about 30 or 40 populations, you find that some populations are completely 100% susceptible, and there are some, one or maybe two, which are 100% of the individuals, they are tolerant. But there are others where there is a gradient of variation with reference to the susceptibility and tolerance. So this is what I'm trying to drive home the point, the effectiveness of sampling. See, those tolerant types might be due to several mechanisms. Tolerance might be due to different mechanisms. So those things you will lose if you go for very low extent of sampling. And if you have collected all those 100 individuals, 100 seeds from a single plant, which is possible in most of the wild species or the even land races, you will be losing out on capturing the type of genetic diversity that you intend to sample. So that point should be borne in mind when you do this type of collection programs, as well as when you go for regeneration, as well as when you go for, let's say, the genetic diversity analysis or any other type of downstream applications. So this particular slide essentially shows you the same aspect, what happens to the allelic diversity when you have a bad sampling done. So in this N, that's the number of individuals sampled is 48 against the 18 individuals sampled. You see that the extent of genotypic diversity that you have captured drastically goes down if the sampling size is very small and it's not optimum. Okay, this point please bear in mind even in your genetic diversity analysis. Whether you are using SSR markers or SNP markers, this has direct relevance. See, why I stress on it is because we have also used a lot of SNP markers, but the logistics or finance that is available to you do not permit analyzing more than one individual per population or per variety. So that's where you will be losing out on the genetic information that's existing in those populations. That's the reason why maybe a lesser expensive tool is more informative compared to more precise SNP type of information, but then it all depends on what exactly you want to look into. Okay, so when you go for measuring genetic variation, see the way it is measured, that's very much important. So the variation of polymorphism can be assessed at different levels of the organization of the that particular population, whether it is individual level or whether it is the region level. So depending on the way you do the further downstream analysis and then the statistical analysis, the type of information that you gather, that you can derive from it, will be different. And then the sampling strategies and size, it may depend on organization of the species. Whether it is, if see, India has, I was referring to Vigna sublobata, it's a native species. So the extent of diversity in most of the localities that you go for collections or where it exists, it is not so much restricted. But compare that with any of the Phaseolus species. See, that's essentially introduced. So the extent of diversity that's existing in the population, whether it is land races, varieties, or farmers varieties, will be much restricted. You take example of maize, even though it's a highly genus heterozygous population or the group of individuals that you are sampling, maybe that type of what differs from one variety to another variety, that variation maybe you can reconstitute with few crosses. If you to quote Harlan's statements, he said using 32 different inbreds of maize, I can reconstitute any type of diversity that you have in maize. So it's also true for other crops. That's easier to do in case of maize. And that's probably is what is being done in case of reconstituting, specifically in case of the linkage disequilibrium type of studies by the Cornell University groups. So that's the reason why in a introduced material, that's fine, good. But in a native species with higher diversity, it may not hold good just going by few sampling few individuals. Suppose you have, let's say the example of chilies, an introduced species, but then introduced several times through several means as a food material or as a germplasm, so on and so forth, or varieties even. But then it has interbred due to its breeding behavior and reconstituted itself, and that has led to people calling India as a secondary center of diversity. So those type of cases, you may have to go for larger sampling. So larger number of individuals need to be sampled. But if it is a crop like, let's say, Rajma bean, self-pollinated, few individuals, only few thousand individuals that have been introduced, or varieties that have been introduced, within a variety, the extent of sampling can be highly restricted. Okay, then the individuals are different geographical origins. If they are if it is so, then the type of sampling you need to follow will be different depending on the type of mating system that is there, so on and so forth. Okay, just to give an idea about extent of genetic variation that might be existing in different crops or the crop species. See, it's estimated based on the basic genomic studies that have happened in all such a wide spectrum of species, that one base pair in every 1350 bases are different. Or so what it means is that 1 in every 1350 base is polymorphic. So that's the, and if the genome size is, let's say, 350 odd in case of rice, you can calculate the extent, 350 megabases. You can calculate the number of expected number of polymorphisms, single nucleotide polymorphisms, that you are likely to encounter. So these type of polymorphisms, if it will be very huge if it is in the non-selective, non-expressed regions. So even though we say that the occurrence of ATGs is basic nucleotides, they are random in the as a whole in the genome, it is not exactly so. It is distributed differently with different frequencies in different regions. If it is a non-expressed region, in so-called chromatic regions, then the extent of variation will be higher. If it is closer to the expressed gene, then the type of variation that you, or polymorphism that you encounter, will be comparatively lower. And in the expressed type of genes, with direct effect on the way the proteins are produced or enzymes are produced, are encoded, then the type percent of polymorphism, extent of polymorphism that you will encounter will be too low. That has to do with the type of repair mechanism that takes place. And if a protein that is functional in terms of an enzyme, if it varies to a very large degree, then its biochemical characteristics will differ, and it may be rendered useless for a particular function, to mediate a particular function. So that gets selected against. That's the reason why these expressed loci, they vary comparatively or drastically less extent in comparison to the selectively or the non-expressed loci. And then the different gene forms that contain polymorphisms that are called as alleles, these alleles you can define in different ways. So usually in terms of sequences, maybe a group of sequence variations or SNPs may be called, it's more sound to call it as an allele if it governs a different type of gene expression, or if it is a completely not non-express or the neutral type of region, then each type of variation, if it is a fragment type of locus that you are analyzing, like in the case of SSR or RFLPs or so on and so forth, then each of those blocks are called as variant blocks are called as alleles. So the definition actually varies depending on the type of gene that you are actually targeting. But what's important is that genetic variation, each type is important, because we cannot at this present day knowledge call anything as a junk DNA as it used to be called earlier. Okay, then coming to the basic forces that shape genetic diversity. You have these four forces, that is the migration, mutation, then the recombination, selection, and drift. So we will be just talking few sentences about each of those aspects. What's most, see, mutation happens in all the populations, but then the frequency is so low, extremely low. It is realized after few thousand generations of perpetuation. That's the reason why immediate effect of mutation may not be so drastic. Further, in addition to that, mutant alleles are always recessive. Even if it is codominant in nature, in expression it is recessive. So that's the reason why this type of variation is not considered of immediate effect in our genetic diversity studies. Migration has a vary that usually we go for estimations, and the recombination has drastic varying on the type of diversity. So is the selection and drift. Now, uh, few words on genetic drift. See, it's a completely random process, and unlike the perception that it operates only in the small populations, it is not so. It operates even in large populations. Only thing is, it is realized, those fluctuations are realized in small populations more drastically than in large populations. Okay, so since it is a random process, repetition of that type of variation in the same population may not occur compared to the other forces of evolution. So, but if it is in a small population, then drift always decreases diversity within a population, because it tends to cause loss of rare alleles and reducing thereby the overall number of alleles in the population. So this aspect must be taken into consideration, and this drastic if the population size is very small, or if it is a very small population. That leads us to whether there is a concept of, you must have read or used also this particular terminology, effective population size. If we have 10,000 individuals in a population and 100 individuals in another population, is the effective population size is 10,000 and 100? No, it is not so. Essentially because effective population, that size that is denoted as NE, small e, that is referred to as the number of parents that are responsible for genetic composition of the next generation, or the individuals contributing effectively towards constitution of next generation. So it is, see, if there are out of 10,000 individuals, 5,000 individuals have same genotypic constitution, then the type of their contribution is similar. So their effectiveness in constituting the next generation is lesser. That's the reason why it is n total number of individuals is not equal to effective number of individuals in a population. Okay, so this aspect we should take in always bear in mind when we interpret the results, because this NE is a more often used statistics, but less correctly interpreted. So NE is generally less than total number of individuals N because of variation of population size from generation to generation. And if there is unequal sex ratio that also affects NE, then overlapping generations affects this, because there will be contribution coming to the next generation from the previous generations, not the immediate previous generation, as it is used in the estimation of this statistics. Then the geographic, am I running over time, Dr. Jyoti?
It is, sir, 1:30, not still time is there, sir, 10 minutes time.
Okay, then okay, I'll just try to hurry up. Okay, then so this what I just like to drive home is the fact that the actual number of individuals in a population is known as a census number, that is not equivalent to effective population size. So this we should take in bear it in mind. Okay, then this probably consequences of a decreasing population size. See, I have just compiled few basic things instead of combining everything and connecting everything, which has direct relevance, and then we generally use in our genetic diversity studies. Okay, so the population size I did say about so much earlier, so I'm just skipping this. Then the aspect of bottleneck. See, bottleneck and founder effect. Generally, these two concepts people confuse. See, the bottleneck develops when a population size drops drastically, whereas the founder effect occurs due to colonization of very few individuals of a new environment. See, the process is completely different. So that's the reason why these two terminologies or the factors must be clearly understood. So in the bottleneck, it may lead to only few due to chance or due to proximity to the next locality, only few individuals may contributing to the next generation. So that's a bottleneck. But if it is, let's say, a few individuals getting transferred to a new locality due to that particular aspect of the dynamics of the population, that reduces the total diversity in the new locality. That's a founder effect. Okay, to quantify polymorphism, there are several parameters that are available. This is just one that is known as nucleotide diversity that you might have encountered or used when you use the SNP type of variations or the sequence type of variations. The way it is calculated, so comparatively very simple. Breeders can easily understand it, so I'm not going to the details of it. Then the total heterozygosity measures based on actually, let's say, the Hardy-Weinberg equilibrium principles. So you have the observed heterozygosity, that is frequency of this type of heterozygous individuals in the populations, as against that what is actually derived or calculated based on the calculated frequencies of alleles P and Q. So that is the proportion of expected amount of individuals of heteros or the heterozygotes in the population. So that's what I was referring to. If this proportion of this heterozygous individuals is lesser than this, it indicates inbreeding. If it is greater than, that indicates preference to cross-pollination due to whatever reason in that particular population. Okay, then what happens if there is inbreeding, drastic inbreeding happening in the population? Then this proportion, it reduces the, by this proportion, it reduces the proportion of heterozygotes in a population, and that gets distributed to the respective homozygous individuals, genotypes. So that is the factor that is actually given as inbreeding coefficient, or FIS. I refers to the individual, and then the S refers to the, let's say, the particular group or the subgroup. So the difference expressed as a proportion of the subgroup's heterozygosity that is expected, that gives you the proportion of inbreeding that's happening in a particular population. Similarly, you have a index known as, given by Sewell Wright, known as the fixation index, that is very commonly used to distinguish the subdivision or the estimate the subdivision of populations, that is known as FST, and that is calculated this way: the total heterozygosity based on the base average frequencies of alleles, and then the subgroups heterozygosity that is estimated based on the genotypic frequencies. So that difference as a proportion gives you the estimate of fixation index. Now, what actually it means, that particular estimate is, if it is too low, that means there is almost no differentiation of the groups of individuals that's happening. And if it is between 0.15 to 0.25, it is considered as a great differentiation of subgroups. And see, this, some cases like it may be still greater, but what's to be borne in mind is that these factors are just thumb rules, not absolute. So what's good in one may not be the estimate may not hold good in case of others. Like what is good for maize may not be good for rice. So 0.15 may indicate very high genetic differentiation of subgroups in, let's say, a self-pollinated crops like mung bean or urad bean. Compared to that, in maize, a cross-pollinated crop, that particular estimate may need to be higher. Okay, this what happens if there is that leads us to the presence of population substructure. So what happens if there is subdivision of populations due to, let's say, this may happen due to fragmentation of habitats. So fragmentation of habitat happens due to presence of barriers or gradients or the differences in the fertility and other ecological factors, so on and so forth. So that population, so that leads to isolation of subgroups. So in effect, the total heterozygosity in that subgroup gets drastically reduced. And then there will be genetic divergence that might be happening due to selection and reduced gene flow and genetic drift that will happen within these small sized populations, as we had seen earlier. Okay, so the genetic drift, the versus how it happens in relation to the number of individuals, that's what is given here. You can see that the number of individuals or the populations where the alternate alleles get fixed, it is very drastic and very high if the total number of individuals is lesser or it is existing as small populations as against the large populations, 100 or higher number of individuals. Okay, then the diversity measures you must be very familiar with. That is the percentage polymorphic loci, number of alleles. These are very simple measures. And the effective number of alleles. Just a word on effective number of alleles. Effective number of alleles refer to the alleles with near equal frequencies. So if we have two populations where this effective number of alleles, in one it is greater, in another it is lesser. So the first one is considered more diverse because each sampling that you do in that population, the number of diverse alleles that you encounter in those samples will be higher. That's the reason why it is considered as a more diverse population. Then this we have talked about, that's the heterozygosity expected, also known as gene diversity or polymorphism information content of a particular loci, and then its comparison with the heterozygosity observed, how it is done. Then the basic population structure as defined by Constantine in 1994. This is in terms of F statistics. It's a very important statistic that's caught up in genetic diversity analysis. Later on, the 2000 onwards, to be specific, and there are different improvements of this particular genetic dive. I'm just skipping. If you, I just want to have interaction, so I'm just skipping many of these. So it has direct implication in germplasm management because it tells you the let's say the type of conservation strategies that you are using, and what happens if it is ex situ or in situ or on-farm conservation, and the type of sample size that you use in regeneration, and the effect of random drift that may happen in those regenerated populations. Just an example of the study that was done earlier in our lab along with Dr. Best of germplasm evaluation division. So we compared the number of alleles that are present in existing in ex situ collection of the same landrace of rice, that is Jolia. And also we collected from the farmers field from Uttarakhand, from different valleys, so many population samples, and did this analysis. You can see that the stars they refer to the gene bank accessions. So the number of alleles is just 1.31 as compared to, let's say, in case of the ex situ collection, 2.56, so on and so forth. So what's important is conservation of allelic diversity is better in case of farmers landrace populations as compared to the ex situ conservation conserved samples, mainly because, see, in some of the groups of these land races, we find that the type of alleles that gain prominence after few generations of regeneration, the whole picture changes. The rare alleles in natural population, they, due to the type of selection process or the inadvertently done selection, that tells on the type of evolution that takes place in the regenerated populations. So if we don't give due consideration to the basic structure of land races, it will completely diverge from the naturally conserved farmer conserved land races. That's what is important to take away from this particular type of study. So that's the reason for that is what is highlighted here. So the main reason is, even in rice, you may go for just 5 or 10 individuals per land race or per accession when you do this type of analysis. So obviously these 10 might have come from 1 or 2 plants that have been sampled in those land races originally. So if that happens, there will be complete loss of the genetic diversity. And that's what actually we found in these studies. Okay, this I'm just skipping population. What I will come to is this particular classic study, which you can always do when you go for genetic diversity analysis. See, this study was done in 1942 by Dobzhansky's group in a desert plant known as desert snow, that is Linanthus. So what they have done is they have surveyed around 30 square mile area, and for each subpopulation, substantial number, 4,000 individuals are sampled. And the what is plotted here is in that 300-kilometer across desert, the occurrence of heterozygosities in each of the sub samples that have been done. You can see that it drastically varies. The demography varies from one region to another region. And in the central region, unlike what you expect, there is a lack of alternate allele in this particular subgroups. So this is what actually is known as the presence of population substructure. So there is very drastic existence of population substructure in this particular open pollinated desert plant. So the way it is calculated, I'm not going to that. The how it is actually interpreted is also similar to the way FS is interpreted. And just a small comparison. See, it varies from the organism to organism, the FST measure. In some, it is very high, the FST measure. In some mammals, it is very high, but in humans, it is a bit low in certain races, and certain others, it's very high. So the reason for that can also be discussed. Okay, so that was FST measure, but there are improvements of the estimates of the FST, which has less statistical, I mean, not significance, it is the quality in it. So to improve on that, the RSC, GST, and theta measures have been brought out by Slatkin and Weir and others. So this is just one example of it, how it varies. So direct Nice estimate of FST 0.23, whereas Cockerham's based on the derivations from the AMOVA, that is very there is drastic difference there. So what is correct, what is wrong, that is debatable. So how it is actually theta or the FST value derived from the sum of squares is given this is in this AMOVA table. So that has come from the analysis of molecular variance done with the very good software known as Harlequin. And it's a good software that some of you might have used, but I recommend that this is better than most of the other softwares and more relevant and gives you very good graphics even. And some other softwares which I generally use, that is the PopGene for basic population diversity parameters. Then you have DNASP if you are using sequence type of variations or the data. And this gives you very good tests for significance for various parameters. So that's just one more slide left for it. In addition to that, the most commonly used the present day workers structure. So what actually it gives is it gives you, let's say, the lower panel here. You estimate the putatively what's the correct number of subgroups existing in your group of individuals that you are analyzing, and then try to see whether there is good amount of variation existing or support for the those groupings that you have done. So in this specific case, you say that see that each color reference to a different subgroup. So if K is 5, you have 5 different colors there. If it is 8, you will have 8 different colors. But this is the plot of those allelic composition of each individual accession that you have analyzed and just plotted as per the putative groups that you have designated with prediction that or the indication that correct group number of groups is 5. But suppose I disagree with this, I'd replot it, the same data based on the genomic affinities or genotype allelic affinities of each of the individuals. You find that there are 8 distinct groups that are possible to be distinguished. This is what I just wanted to tell you about. See, the just don't end up in this. Try to go for the next level of analysis when you are using this type of software, and it gives you very good statistics. And this is comparatively better. Statistical qualities of the estimates is better than others because it's based on Markov chain type of analysis. MCMC is what they call, and the number of iterations that you do is also in tens of thousands. That's the reason why estimate has better dependability. But then don't end with this. Go one step further and try to redo the analysis after regrouping them. That's what just I wanted to bring out. Okay, so I have confused you sufficiently. The reason why I have highlighted these sayings is because see, all are model based and all models are wrong if you go very minutely to the aspects of whether the assumptions are met or wrong, but then they are useful to do. Okay. So I end here. I invite your questions so that that may be more useful for you.
Thank you so much, sir.
Yeah, please.
Myself, Dr. Mamta Singh. Yesterday I called you.
Yeah, okay.
So thank you so much, sir, for providing such an elaborated account of population genetics. And till now, we don't have any questions in the chat box, but if somebody is interested, please ask the question.
Trainees, actually, you are breeders. I don't know how many of you have taken any population genetics courses. See, the present day, when I was teaching in IRI and NBPGR, we found that the number of students opting for this type of courses is extremely low, even in genetics department. That's a tragic part of it, although each one of us, all the publications that come out in genetics and biotechnology or genomics, we see that invariably they use these statistics or the population genetic principles. But whether what we are using, the softwares that we use, they are appropriate, the way we have coded our data is correct, and the interpretation made, that is what is correct or not, that is what is the point of concern when we go through the publications. And most of the cases nowadays it is it's in thing to use the R based softwares. Whatever program that you use, please go through the manual or the original publication, see that whether it has used the right type of procedure to arrive at those derivations or the statistics. And some cases specifically for R based programs, I'm not really very happy with the way it is done. See, you can calculate allele frequency using the Hardy-Weinberg principle, or you can calculate allele frequency by just simple derivation from the total number of genotypes that are there. So each of them has different quality of the derivations that are made. So what is more appropriate will be Hardy-Weinberg principle-based calculations. So please bear this in mind. Any questions that you want to ask? I'll be happy to elaborate.
For participants, I don't see any questions, sir. But I have one query.
Yeah, please.
Sir, in case of biparental population, this population structure studies that can be done or not? And if it can be done, can we divide that population into other subpopulations? Only if it is a biparental population.
Yeah, see, if you go through the literature, Dr. Mahapatra and myself, we had done that type of study using a F2 population in rice. And we did end up identifying a set of SSR markers linked to the trait of interest to his study. It does give you a good result. If there is no other way, you can assemble the number of individuals required for that type of analysis. You can go for it. See, if you go through the present day, let's say the populations that are experimentally generated using best example is in case of maize inbreds, using 32 inbreds crossed to a single central inbred. They have generated a association analysis panel of around 15,000 individuals, which is open for use by any maize worker for further evaluation. See, that has led greater specificity and less ambiguity compared to using, let's say, a core collection of maize. See, the core collection is good if you are sure of the, let's say, the historic recombination events that might have happened, so on and so forth, as it is required in association analysis. Otherwise, it will be more precise, less ambiguous, no background noise in the analysis when you use this type of sequence type of data. If you have a, let's say, experimentally generated population using these significantly identified diverse land individuals or the varieties. See, we know the genetic constitution of each of those individual varieties that have gone into producing these populations or the population diversity panel. So that's the reason why the ambiguity or the background noise will be lesser. You have a provision to, if you use this type of individuals, that is biparental or multiparental populations, you have a provision to select out the background noises from the true signals in your analysis. So there is no reason why you can't use biparental parentally derived populations in association analysis or this type of analysis. And only thing is, take care of the in the model that you use, the kinship and existent of population substructure. So compensate for that, include that in the analysis. And don't go by just the multiple regression type of analysis in associations. Okay.
So,
It gives you, yeah.
what will be the recommended size of the population that we are taking? Because F2 is such a large population.
Yeah. See, F2, it must be possible to generate even 500 odd the individuals in the next generation. So if you can accommodate, if the finance allows, provision allows it, 500 will be a good number, but never use less than 250, because then the angularities of statistics comes in. So 250 to 500, 500 is a good number to use if it is a biparental. But then if the extent of diversity between these two parents that you have used is narrow, then 500 will be redundant number. So maybe 250 will be a better number. So the select type of parents that you use in generating the biparental population, that's important. Okay, so if you are using near isogenic lines with an idea to specifically implicate a set of genes as responsible for a trait expression, then very low number is sufficient. I think what we had used there was maybe 150 or so in our rice work. Even that give a very good estimate. But then but what we had used was SSRs. So the logistics or the implications of using sequences will be different. So if you use a, let's say, haplotype type of data, if you are using sequences, that is a better way of doing this association analysis than single nucleotide polymorphism, because a group of 10 SNPs from a particular gene region may have a correlated response that will have a negative effect on the final estimates. Okay. Yeah, somebody.
So no questions. So thank you so much for explaining nicely presentation as well as questions that I asked. So with this, I am very much thankful to you, and I want to thank you on behalf of our organizing team members also and our directors and NBPGR as a whole for delivering and giving us your important time. So thank you so much, and we would like to hear you in future also, such nice
Yeah, sure, no
presentations.
problem. I'll be sending this PPT to you. You can just distribute it to
Yes, sir,
trainees.
sure.
Okay.
Thank you so
I'll
much.
send a extended version of the PPT,
Thank you.
which some slides I have 50% I had deleted there. So that original version I will send it so that it will be easier for them to understand. Okay.
Okay, sir.
Thank
Thank you. Thank you so much, sir.
Thanks a lot.
Thank you, sir.
Thank you.
Okay, so this is the announcement for our participants. So please join by 2:30. We will start our practical session, which is on bioinformatics tools.
Okay. So, I'm sharing my slide. Just wait. So a very good afternoon to all the participants, and we welcome our today's speaker, Dr. Dhamma Prakash Vankere, and along with him, Dr. Amit is there. He will be joining soon. So both of these speakers will be hosting our session today and deliver the practical session. So first talking about Dr. Vankere. So he is currently senior scientist in the division of genomic resources at ICR and BPGR. And his field of specialization is generation of genomic resources in minor pulses and oilseed crops. And his salient achievements include identification of genomic resources in the form of microsatellite markers in black pepper, then identification of SNPs at genome-wide level using GBS approach in linseed. Then he has also identified various QTNs associated with flowering time, maturity, plant height, seed weight traits in linseed. And also his nature of work includes DNA fingerprinting of pre-released varieties in minor pulses, and he is handling two mega projects, one on the minor oilseed crops and another on minor pulses. And he also has conferred many awards too. Some are here, like SERB Early Career Research Award, in the form of research project for the period of 3 years. He also got the Young Scientist Award in 2016 by High Tech Horticulture Society, Meerut, and also CSIR UGC Junior and Senior Research Fellowship. And he has a very good number of publications in various well-reputed journals. Then let me introduce our second speaker, our senior scientist, Dr. Amit Kumar Singh. He is specialized in the generation of the genomic resources and trade mapping in cereals and pulses. And his salient achievements includes identification of the novel promoter that can be used in the plants, then development of Tino transcript database, which is a database of transcripts and microsatellite markers in Tinospora cordifolia. And he has vastly contributed in the agromorphological characterization of lentil germplasm of Indian National Gene Bank, and also developed a core set. Then one of his most accomplished work includes the development of GDIRT, which stands for Germplasm Duplicate Identification and Removal Tool, which is a very good web server for identification and removal of the duplicate germplasm based on identity by state analysis, which uses single nucleotide polymorphism data. Sir also has conferred many of the awards like Merit Scholarship during graduation, then Kirtkar Memorial Gold Medal, then qualified CSIR JRF and ICMR JRF in 2002 and 3, senior research scholarship for PhD from IARI, and many more, and including RS Baroda Young Scientist Award. So with this brief introduction, I welcome both of our today's speaker, and further I request Dr. Wankhere to please carry forward the session with your lecture, sir. Thank you so much, sir.
Thank you. Thank you. I'll share my slides.
Yeah.
Is it visible now?
Yes, sir, it's visible. Make it in slide mode, please.
Is it okay now?
Yes, sir, okay.
Okay. Good afternoon, everyone, respected sir and madam, for the participants of the training, and thank you, Dr. Mamta, for the introduction. So in this today's practical session, I'll try to demonstrate here how the different bioinformatics tools can be used in characterization of genomic resources, also in generation of genomic resources. So looking at the profile and background of the participant, I have restricted my presentation to all the resources which can be used in the Windows format, not in the Linux or the Ubuntu format, and very basic bioinformatics things like use of NCBI, development of markers, and all. So before directly going to the practical session, I'll just try to tell what is the importance of the bioinformatics here using. We know that since 1990s, the sequence data in the gene bank, like NCBI gene bank has increased exponentially. So we could see here the amount of the sequence which were being submitted in the gene banks, and this is just till 2012, but after that also the number of the genomes and the data have exponentially increased. So we can see the importance of bioinformatics to draw a meaningful conclusion. So in this, actually, the amount of data and the DNA sequence, protein sequence are there abundant, and our job is to find out what useful information we can draw from the available data to solve the existing problems of our research. Now, we know when we have very large data or very large information, it is sometimes very difficult to interpret in all its perspective, in the holistic way. So if we look it in the narrow way, we may have a wrong interpretation and the wrong conclusion in the science. That is why a holistic approach of this is important, and this picture represent the same. So now this I will come to the practical session now. Suppose so in the practical sessions I have limited on the two aspects here. One, development of SSR marker, because we know SSR markers are the markers which are preferred by the researchers, plant breeders, which has also applications in the marker assisted breeding. So that is why I chose here the development of SSR marker from the limited resources. Now we know that SSR markers are the codominant markers, and it is the repeat of, like it is, these are the simple sequence repeats, the repeats which start from dinucleotide to hexanucleotide. So such repeats in the tandem way, if it is occurring, that we can call is simple sequence repeat. And the variation in the repeats is the marker, and that it is known that these repeats are highly variable. And it is also important to note that the adjoining sequence, the flanking region of these markers are often conserved. So here in the practical session, although I have used only the screenshot, but if required, I can demonstrate also in this. So here we what resources we'll have to start with. So I have used an example of Dolichos bean here, like Dolichos bean, lab purpureans. Suppose so we wish to develop the SSR markers in any underutilized crop, or you say potential crops, and we do not have the resources to start with now. So I have used these references. So we know that there are a lot of ESTs in the past, in the last decade, and the transcriptome sequences and the reduced representation sequence have been deposited in the repository like NCBI Gene Bank or the SRA archives and all that. So such information we can make use to identify or the develop SSR markers. So the example here is lab purpureans. So to download these ESTs, download this bulk EST, we can go to the NCBI. I hope all participants must be aware of this NCBI database. Here we can search here what we want to search for, like whether we want to search for nucleotide or some other database. So I have chosen nucleotide, and then we can select a plant and then search. So we'll find out, we'll get the number of the hits here. From that, now since the nucleotide database includes EST sequences also. Now, so on the left side, you will find the different types of the hits it is received. So if we click on what we want, like I have clicked on the ESTs here. So whatever the hit I have received now, around 1686 hits. These hits are only the ESTs which were developed and deposited in this NCBI. Now we can download all these ESTs in the bulk. So to download that, we can go to this send to. So since this is a slide, I won't be able to show it here. When you go to send to, and there will be a send to file. So in that way, you can download all these ESTs in one go, all these 1600. Now these downloaded resources can be used to find out what SSRs repeats are there in this data, in this downloaded sequences. Now I'm using three different database, or you can say resources, to develop this SSRs. So there are a few sepals, and then the complexity little increases. The inclusion of all these three I have purposefully used here, although the purpose can be solved in any one of these three resources, but it will be helpful based upon the requirement And also this will help us to learn more things while I go through this. So now this first resource is simple SSRIT, simple sequence repeat identification tool. This is available in the Graminy database. So we can search that and find out this SSRIT tool. I have given the link of this tool in the tutorial also, which I think all the trainees must be having this tutorial with them. So clicking on the link, you can go to this database, Graminy SSR tool database. Now, once it is open, you can you have to paste your sequences here. Whatever the sequences you have copied, you can paste the sequences here, and then you select what kind of repeats you want. Do you want dinucleotide, tetranucleotide, pentanucleotide repeats? So if you put here pentanucleotide repeats, all the repeats below pentanucleotide like di, tri, tetra, penta will be there. So here we can choose, suppose we want to identify all the repeats. So we can use maximum decamer. So it will include all that. Now, how many repeats we want? Like for a decamer, at least if it is repeated 4 times, tandem repeated 4 times, we can use 4 here, or we can change also 3, 6 based upon our complexity, what requirement we want, we can choose. Then paste our sequence and then find this SSR tool. Now this will this is the second thing, like down screenshot. Now, after entering in the repeats and pasting the sequences, you just find SSR. I have used only one sequence here for the for demonstration. So it will, if it has any SSR, so it will show here what motif is there, which motif has been identified. Like here, it is a TTC, and how many times it is repeated, minimum 4 times. What is the start site and what is the end site? What is the total sequence length? Okay, that is there. So in this way, we can find out SSR. Now, the finding just won't solve our problem. So we need to design a primer for that. So to design the primer for specific SSR, amplification of the specific SSR. Now there is another tool for designing the primers. This is called a Primer 3 web tools, web program. It is freely available. And the link also is given in the tutorial. So here we, so here we can paste our sequences. Now we want our primers around this SSR repeat. So we have to mark this repeat with the bracket sign, which I have highlighted it. So that the primers can be designed on primers will be designed flanking this selected region. So the program will identify that this is a bracket and this is to be identified. Now there are different parameters which we may give if we want some specific things. Like the target thing I have mentioned here, that these are the target things. And if you want to exclude some regions, like you do not want a particular region for designing the primers, like in any extra repeat, AAA or TTT repeat, so we can exclude that region. Also, we can mark that region. If you want to mention the start codon or any internal oligo regions, or if you want any overlapping regions, all these things we can mention here. And then you select the primer. You can just pick the primers. Yeah, this also can be used for selecting the hybridization program for the hybridization, selecting the hybridization probe in the northern or the southern hybridization, which which were used in many labs. So we can choose whether left primer we want or the right primer we want or both the one. So based upon that, the primers will be designed. Now, going little down, there are additional parameters. We also can select what is the size of the primers, how many our primers, what is the number of nucleotides we want in the primer. Optimum size, what is the extreme? It should not go below 18 or it should not go maximum, whatever the required number. And what is the minimum TM or the TM range we want? Because the forward and the reverse primer should not have much variation in the TM, because we will be using both the primers in the PCR. So the TM should not vary much. So we'll keep here that the TM is this much. Then this is primer TM, product TM is doesn't matter much. Primer GC content matters, and that region we can select what is the GC region, how much maximum it should be there. Anything beyond beyond 30 means minimum 30 is good. Then what is the product range we want? So product range we can give here that I want just 150 to 250. So we can exclude all that and we can limit these things that much. And while designing primers, it is also important that both the primers should not form a dimer and by pairing in themselves. So that is what we need to find out, what is the thermodynamic secondary structure if they form. That restrictions we need to keep. Okay. Keeping all these things in mind, then we can pick the primers. And the primer 3 output will be in this way, like left primer and the right primer. Here it will give the most the primers which meet all our mentioned criteria. That will be given the best primer here. So in this is our region which we selected for designing the primer. Like this should include, and this arrows indicate the forward primer, and this is a reverse primer. And now these are the primers, left and right primers, and what is the length of that primer, what is the TM in that primer. So as I mentioned, that if the TM is this 59, 58, this is quite good. What is the GC? It's beyond 35, so that is quite good. Any thermodynamics like secondary structure, hairpin structure, nothing, it is not forming any. So this is the ideal situation where there are no secondary structure or hairpin formation, so that the primer do not make a secondary structure and do not form a dimers, and we'll get a successful output. And these are the primer sequences. In addition to the best, the primer 3 also gives another 4 to 5 sets of primers which are best, and we can choose from that what is best suitable for us. So Dr. Mamta, in between, if anyone has any query, they can ask, or should we take at the end of the presentation? So if anyone has any query regarding that, can be asked while making presentation.
Sure, sir.
So, okay, so in this way, we can design the primers for individual amplifying individual SSRs. But we know that generally SSR primers, we do not go for the individual SSRs, and we go for the bulk or the more number of SSRs. So there is another tool called WebSat tool. It is web software for microsatellite marker development. This is relatively easy. This is relatively easy than the earlier one.
Soft car
So here we can, this is the link, we can open this link, and here we can select what types of repeats we want here. It is up to hexanucleated, we can choose here, because this is by definition, it is died to hexa. Generally, the mononucleotide repeats are excluded in identification of the SSR, so we can exclude that repeat. We can select nucleotide repeat type, based upon the type of repeat, how many minimum repeats are required. Like generally for the hexa repeat having the 6, we may not get that the number of repeats, so we can reduce the complexity here. Like hexa repeat can be kept 3 to 4, penta can be 3 to 4, and the di, tri can be more. So selecting this, pasting our downloaded sequences, and we submit and we'll get the result. We can also choose a file, a FASTA file, upload the FASTA file and the submit, we'll get the result. So here we get the result in this form. This top sequence does not have any SSRs or the SSRs as per our mentioned criteria. This sequence has SSRs. This blue region, highlighted blue region, is a repeat. It is a ATT repeat. You can say here ATT repeat, and these are the flanking primers. This highlighted in the green are the flanking primers. Initially, we just see a yellow highlighted region in the result. If you click on this region, you will get the SSR and the flanking sequence, and this is the primary information, primary information. So for individual, either can copy or we can export this to the Excel file also, and the way it is mentioned, this is a screenshot of that. So here it will show what is the sequence ID, what is the repeat, how many times it is repeated, what is the forward primer, what is the length, what is the TM, okay. And this one is the product size. What size of the amplicon we'll get when we have this primers in the PCR. So this is the second tool, quite easy tool that can be used for identification of SSR and development of test markers. Now, the third tool which we can use here, the link is always also given in the tutorial, is a SSR identification tool and primer designing using the batch 3 primer program. So this is a very good tool. We can use large number of SSR markers, SSRs here, and can design the primer in the same way. Now this is available on the earlier used to be available on the 2 resources, and now this is available on the USDA ARS server, so that once we open this link, you will open this page and then you will have to go to this server, USDA ARS server. So once you open that, then this it will open this window. Now in this, like here in the choose primer, I have taken the screenshot keeping this drop down menu open so that I can explain the things. Like we can use this for generating another like other PCR primers also, like for generic primers. Here we can select the SSR primers that can be used for hybridization probe or flanking SNP, single base extension, allele specific primers, and for sequencing primers. All these things can be done here. We can paste the sequence here, or we can upload the sequences. We can pick left and right primer. Any mispairing or repeat information, if it is there, that can be given here. In down, we can choose what type of repeats and how many such repeats we want while selecting the SSR. So once you give all this input, you will have such results here. So sometime it may happen that for certain SSR, you do not get appropriate primer. So this is the that case. So we do not get appropriate primer. Now in this case, for such repeats, we get the primers and all this information. What is the product size? What is the GC, TM, length? All this information is available, and this can also be saved in the Excel file. Different these things can be downloaded, and we can also use the analysis portion here, that what kind of total repeats are there, and all that analysis can be obtained from here. These are all Windows based program for development of the SSR primers that can be used by all those who are all those who know the basic computer knowledges. Other than that, for bulk, like for the genome-wide SSR analysis, like we need to identify SSR throughout the genome, and the genome size is very high. We need assembly for all that thing. There are Linux-based or program where the knowledge of some programming language and the scripts is required. So there is a tool called MISA tool, SSR identification tool. So in most of the paper where the genome-wide SSR identification, such things are there, that generally use MISA tool. I did not include that here, seeing the, like I limited myself to the Windows-based programs only. Now, now, how can we make use of the another part of my presentation is in what way we can make use of the available tools for making maximum use of the genomic resources or the sequence information we have. Now, this is, I think most of you known about the basic local search alignment tool, that is BLAST tool, which generally we use in the molecular biology to identify that what the sequence which we get, that is hitting to what sequence and what is the function, possible function, and the functional annotation it has, all that thing we know. These BLAST programs are used. So there are mainly 5 types of blasts are there. One by one, I will go and explain all that. The basic is the nucleotide blast, BLAST-X, T-BLAST-Gen, and the protein blast. There are some other versions also, which I'll explain. Now, when we open this tool, now I hope all must be aware that this is to find out what sequence, the sequence which we get, like suppose someone is using an RAPD primers or any dominant markers, and they have got some band which is associated for some trait, or just want to check if it is associated with some check. That bands can be eluted and the sequenced. Now, such sequence, what is that sequence? What for that we use this tool, BLAST-N tool. So this BLAST-N, a nucleotide blast, uses nucleotide as a query, and it searches against the nucleotide database, like it searches against the nucleotide. So we'll get what DNA sequence is there. That is what we'll get information. So we have to put our paste our sequence here. We can upload the sequences also by choosing the file. We can paste multiple sequences here. Suppose there are 50 sequences together. That need to be in the FASTA format. FASTA format means that should be a greater than sign should be there, and one line of description, and from the next line it should start the sequence information we have to paste here or choose. And then these things actually in the choose search menu in the database, what database we want. We just want the nucleotide collection total, or we can, there are different other resources, like suppose we want ESTs, it should be searched only in the ESTs, or we want to be searched in the transcriptome sequence which is available, or certain genome sequence which is available, or certain shotgun sequencing available, or the other sequences like sequence read archives which are submitted. That thing we can specify here, and we can also specify what organism we want. We want an Arabidopsis, or we want rice, or whatever the genome is available, it can restrict it to that. Generally, people generally we just paste the sequence and then search, and then we get hit. But we can specify that so that we'll get the best possible hit. Okay. All right. So as I was mentioning, this drop-down menu is there. So nucleotide database, reference sequence, some RNA sequence, whole genome shotgun, ESTs, SSR, sequences, archive, generally all this transcriptome or the GBS sequencing, all that thing are archived, are stored in the sequence read archive. So we can select that whether our hit has any sequences matching to that or not. Okay. So all the things can be obtained here. So selecting the database and organism, we can get the specific hit. All right. Now other type of blast. So now this generally is used to just see what the nucleotide match we get. So suppose sometime we had just kept the nucleotide collection as a sequence and we had done some match. So it often goes and hit to the chromosome of the respective organism or some scaffold sequence, and it just gives information that this is the scaffold of this Vigna radiata, Vigna mungo, the chromosome. So we know that it has given the hit, but doesn't answer many of our queries that what the function it may have. But it will just give an information that there is a hit and it is more similar to what organism. Okay, so that thing can be there. So what organism it is most similar. Now to get more detailed information about what possible function it has. Okay, so there is another blast is called blast P. So blast P generally uses, not generally, blast P uses protein sequences and it searches against the protein database. Okay. So if we have got a protein sequence somewhere with us and we want to see what is the most closest ortholog of this sequence, sometimes we need to find what is the best ortholog of our protein with the other organisms. Suppose so that we can find out what function it has and whether any functional information is available about that whether the knockout mutant and all such any such paper published or not. So protein sequence generally are used to select to find out the ortholog. So blast P is one of the methods to search the orthologs in the other species. Okay, so in this also you can select different databases here. Okay, a particular organism or the protein sequence of the UniProt database. This also can be used to see whether our protein sequence has a protein sequence has an ortholog whose the 3D structure is available or not, the similar 3D structure. So this particular thing, protein data bank. Okay, so PDB, this is PDB. So PDB shows hit of the sequences which is having 3D structure already available. And when I say the 3D structure of this protein, it is only either through X-ray crystallography structure or by the wet lab methods, not through homology modeling. Homology modeling sequences are not available in the protein data bank. Only NMR based or extracrystallography based sequences are available. Okay, so that can be found out. So what application now, why will someone search a 3D structure? Suppose you want to see what are the possible interacting partners of your protein. Suppose your protein is a transcription factor or your protein is a kinase, and now this kinase is going and binding to what protein or what transcription factor. So for that we can find out whether the 3D structure is there, and using the different model we can find out the protein interaction also. There are different tools which are available that can be found on the protein interaction pattern. Suppose sometime an interesting thing, a host pathogen interaction. Suppose an effector, it is effector protein is interacting with the resistant protein or not. To find out such interaction, protein structure are required, and in that way we can find out this structure, similar structure. Okay, so the another type of blast which generally were used to find out what is the function of the nucleotide. So in this we use nucleotide. Okay, and then in this the nucleotide is translated by the program, and the translated nucleotide is searched against the available protein, searched against the protein database. So here the query will be a DNA sequence, but it will be translated, and the hits, whatever the hits we'll get, the hits will be a protein sequence. So generally when we want annotation, gene annotation in that, and we do not have the protein, like suppose we have ESTs, we do not know what is the start size and what is the stop size. So what frame it is there, we don't know anything. Okay, so in this case, we can use the BlastX program. So so that it will translate in the multiple frames. It translates at least in five frame, I suppose, and then that searches against all the protein sequences, so that we'll get a functional annotation. So generally earlier EST's functional annotation were used to be done by this BlastX program. So that can be used, and here also we can use different databases, what organism we can use, and all that. Now another BLAST is T-BLAST-N. So in this, we have a protein sequence. Okay, that protein sequence, it searches the translated nucleotide. The protein sequence searches against the protein sequence is a query. It is retranslated like to the DNA sequence, and then it is searched. Okay, the protein is the query. Okay, and that is converted to the DNA sequence, and then it is searched against the nucleotide database. Okay, so we do not have, this is used when we do not have nucleotide sequence, we just have the protein sequence, and then we find out what is the nucleotide, possible nucleotide sequence, and what frame it is there. Now the another BLAST is this last one is, sorry, the last one is this T-BLAST-X. This I did not click it here. T-BLAST-X. So in this, the protein sequence, the database is also translated protein sequence, and the nucleotide is also translated protein sequence. So this is the most slow BLAST, because it translates all available sequence, nucleotide sequence, and then translated into translated nucleotide, translated, and the translated database. Both query and database are translated. That is what T-BLAST-X. And here also we can use different these things. Now in the BLAST-P, I have missed to inform you. Like suppose our objective, there are different algorithm also for using the BLAST-P. So suppose we want a iterative BLAST. Suppose we want to identify the distant ortholog or distant homolog. Suppose I have a plant sequence and I want to identify what is the possible protein in the human being or in the mouse. So for that, like this PSI BLAST can be used. Okay, so position specific iterative BLAST. So what does it do? It does an initial BLAST, and then whatever the queries we get, so it makes the consensus of that, a profile of that is prepared, and then that is again the BLAST is done. So again, whatever the result we get, again from that a consensus is prepared, and BLAST is done. So the iteration of that is done, and that is why it is called as PSI, position specific iterated BLAST. This will identify the distant homolog, what function it's in a distant organism. Then there is the 5 BLAST, what specific pattern hit we want, that can be obtained, or certain domain. If we want to look only a specific domain in the protein, like in the transcription factor, I want to focus only on the DNA binding domain or the maybe interactive domain. That can be in that case, this delta BLAST can be used, right? So any queries in that? So I have come to end of this presentation. Any query, if it is there, I can solve, showing the practical demonstration to them.
There is no query, but Dr. Hitesh, he has requested to share these PPTs.
All right. PTNK I can share, but in the tutorial already it is all the links, all the screenshots are already given. So since I have some 10-12 minutes left, I can. Is this screen visible, Dr. Mamta?
Sir, I think you have stopped sharing the screen.
Okay. I'll share the screen. Is it visible now?
Yeah, it's visible.
Now, initially I had told that how we can download all these ESTs in one go. Like I have searched here, Dolichos bean lab perpedians. Okay. So we got hit here. We got different organism. We got this Vigna radiata also. So I have to click only this lab perpedians. Here we click, and then only that sequence will get. Now here ESTs we got. Now to download all this together, we have to go here, send to. Okay. Now here the complete record if you want. So we choose a file here, like send to file. We want a complete summary, or if you want to use directly, then we have to choose here this FASTA. So the FASTA sequences, and then any specific order you want, accession number and all, or in the default. So you can choose according to that in what format you want, what modification, date of release and all that. So based upon that, you just create this. So this will ask the target where it is saved. So we can save. So in this way, we can save all these, like all these 1600 sequence are downloaded here. Okay. So these are downloaded here, and we can open that here. So all these sequences are downloaded here. So now these sequences can be used in the programs I have mentioned for identification of SSR. So in this way, we can download this sequence. We can download protein sequences also. Okay. And while doing the blast, I'll do one blast here to show. Yes, the blast. Okay, here database, we can select all what database we have, as I mentioned here. Okay. Now this we get this window here. So this is the basic description, and then in that, what is the total square, what is the query coverage, whatever the query I had given, how much is covered, what is the e value, what is the percent identity, and what the accession number it is showing hit. This also show the graphic summary of in what way the query has matched to the these things, and these are the then specific alignment, what sequence we get here, and then this is the taxonomy of whatever the hits we got here. So in this way, we can do with respect to blast X, like in this we can, by clicking here, we'll get the FASTA sequence here. Okay, this is cDNA. Plastics we'll see. I'll keep the database just like that, non-redundant database. So it takes Blastix generally take more time than the nucleotide base because it has to translate. In another tool, by the time it opens, there are some alignment tools also like cluster W that can be that can be used to see the alignment uh, between the two sequences and how much similarity it is there. We can download the sequences here, like suppose the blast is done here. I'll just do the nucleotide blast here. Okay. Now this blast is there, then we have to like to find out what sequence which is not close. Now I can download this particular sequence, plastid genome, like going to this sequence ID. I download this particular sequence ID plastid. I can download. Okay, and then to take faster format. Then that can be used. Okay, this is complete. Like I told you, nucleotide blast generally goes and hits to the genome. So now this EST is belong to this plastid genome. So the complete plastid chloroplast genome is there. So now good. Now in this case also, suppose we do not want the complete chloroplast genome, and we want to download only the sequence which is matching. That also can be done. Like in this, we can show only the match sequences. I'll just show here the sequence it is matching. This. Okay, here we from this like we want to identify what is the gene, what is the coding region of the hit. So that can be identified by clicking here, like the series, what is the coding sequence into that. So that sequence, series sequence, we can identify the sequence. It will come here, but the faster sequence can be obtained from by clicking here. So we'll get only the faster sequence of that particular series. Okay. Suppose we want complete gene from that. Here annotation is there, complete this is series, complete gene. If we want what genes are there, so that will be highlighted. It also includes some UTR region, and again you take the blast of faster of that, and that can be downloaded here. Okay. So about the different thing like in tRNA, CDS, all that thing can be obtained using this. There are some in NCBI I also would like to tell, because now the next generation sequencing. So different bio projects are also available. So we can use download the nucleotide or the transcriptome sequence, and that can be used for validating certain genes or validating certain genes, or say the match we have, or checking the expression of our genes or the gene families. That can be done in this. So this sequence read archives. So this is sequence read archives. We can download here. So I'll just type here. So in this, we can identify all the archive sequence read archive sequence, and the transcriptome sequencing database on this. Or sequence of any sequences, so that we can make use of that to have our specific queries address. Okay, so this is here. From here we can download these sequences. All that thing can be done. Right. Okay. So I think done from my side, Dr. Mamta. If any queries, I can answer.
Queries I don't find,
Okay, okay, all right.
But on behalf of all, I want to ask if there are any courses or part-time courses that are suitable for professionals, because they cannot get the time to learn all
Okay.
these things. So
Mhm.
if you know, and
In the like, yeah, there are several trainings which are being organized, online trainings, or there are some courses in the Coursera. There is in the Coursera there are some specific courses actually which are available. Suppose for this basic bioinformatics or some R, learning some specific R programs. All these things are there, and these are very basic things, and there are several videos also, even for the Linux and working on the Linux platform, or MISA, if you type using MISA and working on the Linux identification. Such things are available in the YouTube also. There are several these things. And the Coursera, in the Coursera, there are several courses. Specific.
So what's the average length of these courses in terms of duration?
That certainly varies. It could be few weeks to few months actually. Generally, they organize the like sessions on the weekends or in the off time. So only one or two, maybe two sessions in a week. Generally, it takes time. But yeah, for a few weeks to few months, these courses are available, and some of them are free and some of them are paid also. So that can be searched.
I think that's a good information for those at least who are interested and want to explore more in the field of bioinformatics. So, thank you so much for elaborated explanation of all the tools. I know that 1 hour class is not sufficient for explaining
Yeah,
all.
yeah. I wanted a demonstration actually, but that
Yeah.
would not have been possible. It would be good if it is the live demonstration or the physical training. That would have
Yes,
been
sir.
more.
We also actually wanted the same. Otherwise, this would have been more beneficial for all of us if it was a demonstration in terms of hands-on training. No? So, but anyways, this was a good presentation, sir, and thank you so much. Sir, can you confirm if Dr. Amit has joined?
Yes, just right.
Because I don't find his name.
Oh,
Okay, sir.
maybe.
Hello, sir.
Hello.
Hello, sir.
Just a minute. Hello. Yeah. Okay, I'll change
Actually,
it. My son is using sometime this one, so it's
yeah, actually, I was searching with your name. That's why your name is coming in with different name actually. Yeah, now it's okay.
okay. Okay. Thank you.
Okay, sir. So I
Afternoon.
welcome you for delivering the next part of this practical class.
And thank you, Namta. Thank you. Thank you for the
Thank you, sir.
introduction.
And please share your screen. I already have introduced you.
Okay.
Probably,
Thank you. Thank you.
probably you didn't join by the time.
I had joined that.
You had joined. Okay.
Oh, I was able to.
Okay, sir.
Not able to say. Oh. Why it is not visible? Just a minute. I'll be sharing the screen. Why it is. Should I open this? Okay. Okay. Okay, just I'm sharing. Okay. So now it is visible.
Yes, sir, it's visible.
Okay.
Just make it in slide mode, sir.
Slide mode. Okay.
Yes, sir, now it's okay.
Okay. Thank you. Thank you, Mamta. So good afternoon, everyone. So in the second section, I will be talking to you on genome-wide associative study and germplasm duplicate identification. So you must have already taught about little but theoretical aspect of it. So this class will be mostly on practical aspect, that is the principle of genome-wide associative study, and also we'll be talking about one tool which we have developed, and that is that is germplasm duplicate identification tool, which can be used to identify duplicate germplasm in gene bank collection. So gene bank collection maintain large number of collections of rice, wheat, or whatever plants. And so sometimes it becomes difficult for individual to distinguish them based on phenotypic traits. So since the technology is evolving and we have now new advancement SNP markers available, which can be deployed. So we have developed that tool. So that will be also shown to you, and you can have a practical training on that, and we'll be giving you some instruction. You follow that, you'll be able to learn how it can be performed. So so this class will be taken by myself as well as Dr. Tanmay Kumar Sahu. He's working as project scientist here at NBPGR. The first part will be by me, that is basically theoretical aspect, and the second part purely practical, which will be shown by him. So see this slide. So something comes in our mind, because this class is very heterogeneous, people from multiple discipline, maybe somewhat people not related to hardcore of genetics and genomics will be attending this class. So bringing curiosity of this particular class, I try to take this image. So if you see this image, you can see that there is a variation in plant height of the plants which have been shown here. So ultimately it comes in somebody's mind, what is behind that? What is the factor? What is controlling it? So now it is almost 100 years have gone. Mendel has given his own what is factor concept, and then it all Mendelian inheritance. Now, if general people know that it is what we see today, what phenotype we have, it is basically because of gene. So that is one individual color will be red, some individual color will be white or whatever, in context to animal or in context to plant. So what guide these variations? What particular factor guide these variations? Because in rice, the plant height would be very in a like range. It may be for 100 meter, 200 meter, whatever. So what is that factor which guides? So it is all guided by changes at the DNA level. So that is sequence level polymorphism. Sequence level polymorphism can be simple nucleotide polymorphism, or it can be insertion and deletion of subsegment. So we all know that every individual has a DNA of like 100 rice variety. If you try to study, all 100 may have differences. Some or other point, their genome can be 99% same, but their 1% or 2% of the total content may be varying. And that is due to SNPR index. And that variation may be actually responsible for the variation which we physically or morphologically we see in those plants. So in this particular class, particularly you are interested how to link that variation with the trait. So how we can identify that particular variation, which is actually responsible for differences in the plant height, differences in the seed shape, differences in the seed color. So to find that, now we use different approaches. So there are two approaches. You know, one approach is called biparental mapping, another is association mapping. So biparental mapping, now it is known as a classical mapping approach. So this approach is being followed from last 30 years or so. So where you identify contrasting individual, big, large size seeded plant variety and then small seeded variety, and then cross them, generate a mapping population. And then you generate a F2 or you generate real population and try to associate recombination frequency, what is that, with the trait. So that is what we have been doing for before that the GWAS concept emerged. So we are only using this approach to map a gene. This approach is good. It has enabled identification of many genes. But nowadays we have another approach also available that is called association mapping. This approach doesn't require mandatorily generation of biparental population, just like in case of QTL mapping. Here you can use natural population, or even multi-cross, multiple generation crosses also can be used, but natural population as such can be used. So you can just take out some germplasm 200 from gene bank and then evaluate them for trait and try to associate. So what is the advantage of this approach over biparental approach? Biparental approach, what we do, you are deliberately attempting one cross. So even first generation, we get F2, and F2 recombination frequency is being used to associate and map particular gene. Or if you have a reel, it goes to F7 or F8. So in this approach, since a single recombinant event is happening in F2, single generation, then you don't expect mapping resolution to be high. You have the genotyping data, you have the phenotyping, those molecular biology and genetics may be able to understand. You have generated genotyping data, phenotyping data. When trying to associate, F2 population will give a mapping, but its resolution will be poor, means you will be very, very distantly, your identified gene may be very distantly placed to the associated particular causal gene. So that it may be even up to 10 MB interval sometime. So the resolution is poor. But when you use concept like association mapping, you use diverse individuals which have accumulated recombination over the generations. So number of recombinant events are numerous. So that aspect you are taking, plus you are taking multiple individual, and then you are also taking advantage of diversity in those individuals. So diversity and number of recombinants being used to map the particular trait. So here resolution will be definitely high because the basic raw material being used have captured more number of recombinant event. Means genome has reshuffled over the year. Like here you see the genome, only two colors are there. You cross blue and then you have a part where blue and red are getting mixed. In this case, you see that over the year recombination activity, multiple individuals, and then you collect the individual from different places and see that the whole genome has been broken in different parts. It means there is a recombination which has taken place. So you can have a marker on each place, and that marker can be getting associated, and that marker itself may be in a self-contained LD block region. So then, so you just now understand association mapping doesn't require mapping population, and it can give you high resolution mapping. Even you can land up to directly the particular gene which is responsible for the trait. So this association mapping can be again divided into two types, that is candidate gene based association mapping and whole genome based association. The basic principle of both the mapping approach is same. Only difference is there. Here we perform on candidate genes. Suppose you are trying to want to perform candidate gene avas in rice, then we'll take already 4 or 5 genes which are known to be involved in grain size determination and seeing the variation across 200 genotypes or 1000 genotypes, polymorphism in that particular genes. So we have taken 5 genes, we have analyzed variation in those 5 genes only and trying to establish a relation genotype to phenotype. So the variant polymorphism, you will limited that you will be identifying using this approach, you limited to particular candidate gene which you have taken. Basically, it is a kind of allele mapping, allele mining approach, where you are searching new alleles which can be controlling grain size or any other trait. So this is only fraction of the genome we are handling, maybe 2 gene, 5 gene, 10 gene. So this is very like cost cheaper approach. And it is a directional that particular genome we know and just try to find within those particular candidate genes which are the particular SNP guiding particular trait. But when you go for whole genome-wide association study, there it is a very comprehensive approach. You are screening entire genome variant, which is SNP, you generate for all the chromosome. Like in case of rice, you generate for all the 11 chromosomes. For each chromosome, you have a 10,000 SNP. Like you generate 1 lakh SNP, 1 lakh 10,000 SNP, and then 1 lakh 10,000 SNP is now looked for association with the phenotype. So variant traits were throughout the genome are scanned for association with the trait, that is whole genome, name itself is a whole genome, not limited to candidate. And now it is possible to identify all the variants where you already known gene, you are just trying to find other allele. Here you can find new gene as well as new allele, because even de novo genes will be discovered which are controlling the trait. In case of candidate gene, you known gene, which you will be only able to identify known new alleles. Here you will identify known gene and even unknown gene. And for known gene, you can get new alleles as well as previously known alleles. So but the approach is little costlier. All the labs cannot afford. Those having funding support, they can genotype large collection site, large collections of accessions in different crops, and can then phenotype, because phenotypic requirement is also very crucial and important, where you have to generate multi-location replicated data to perform association study. So then coming to our aspect, which are here we are dealing only genome-wide association study. So genome-wide association can be defined as scanning the genomes of multiple individual of a crop, maybe human or any animal, it can be performed, and to establish relationship with the traits. So, and then what you are screening in the genome, we have a landmarks like SNP, you have copy number, which indels you have identified in those individuals and trying to associate. Nowadays, in sequences possible, you can sequence 100 individual end to end. All the whatever 300 and whatever 3000 gene you can sequence, or 3 MB size of the genome, you can sequence 10x time. And each nucleotide you can look for their association, each nucleotide of the individual. Means if you sequence rice, 200 varieties on a 20x coverage, then your sequence, you will get each SNP detected optimally, and then that particular genotyping file, resequencing file, you can see. There you get highest resolution using this approach. And then you already told it can be done on natural cultivars or natural population cultivar magic. And then what we get, you get whole genetics deciphered, like it is done in QTL mapping. You know what, how many candidate genes are governing that trait, what is their distribution, and what is their effect. Are they positively controlling the trait or negatively controlling the trait? So those things you can understand. So we just gave introduction and overview what is association mapping. Then what is its basis? Its basis is a concept called linkage disequilibrium. Linkage disequilibrium can be defined as non-random association allele at different site, different locus in a population. So this linkage disequilibrium term is in the context of a population of individual, and then you bring the different locus together, and they see their association pattern. If their association is non-random, then you say that those particular loci are in LD, linkage disequilibrium. And the linkage disequilibrium, if they are there, that is a prerequisite for marker which you have identified, and prerequisite if you identify a marker, and then you can relate with the particular Kajal gene which is present nearby. So just simply understand, LD is a concept in a population, and with respect to 2 or more loci, and you're trying to see the association of those particular alleles in those loci. So here I have just given an example. If you consider 2 locus in a population of rice, locus 1 and 2, locus 1 with an allele A and small a, and then similarly locus B with allele B and B, then if there is no linkage, if it is a linkage disequilibrium, you expect that haplotype frequencies to be product of their individual frequency of those corresponding alleles. So for a particular, these 2 locus, you can have all 4 different type of haplotypes. For each haplotype, you have to calculate what is the haplotype frequency and whether it is equivalent to the corresponding allele frequency or not. And if it is not that one, then you say that then particular 2 locus are in linkage disequilibrium. That is a requirement of finding associated marker and then corresponding gene. This what I was saying, then when you have 8 varieties of rice, then you have genotype them, or you sequence them. When you sequence all the alphabet, you will be able to see. When you genotype, you only restrict to detecting few markers, like whatever given in star. So genotyping with 50K rice chip. What does it mean? We are genotyping only 50,000 SNP in the genome across multiple individuals, across, in this case, 8 individuals we are detecting. But when you are resequencing, we are sequencing all of them, and in each position you are able to detect SNP. So you can use resequencing data, we can use genotyping data. So genotyping data, it will be more a kind of indirect indication of presence of a gene. So if you find that, suppose there could be a particular gene that is causal mutation responsible for seed size. And when you perform association with a genotyping data where only star marker available, then we get one particular star closest one getting associated. Then we see flanking region whether there is a gene or not. And how much we see up to link LD decay distance. So here is here. LD you can see below here. LD is maximum in the vicinity. And as we go away from that causal organism, LD decays, means those markers they will not be again inherited together as a haplotype. So you will be able to identify candidate gene only if you have a highly dense genotyping data. So you will be able to reach, and that's why in case self-pollinated crop, you need to have more and more of marker to generate genotyping, because and in case of cross-pollinated, you need to have more and more, because the LD decays very fast. Each interval one must have a marker. While self-pollinated, you can, because LD decay distance block, because one marker per LD decay distance is at least minimum requirement to perform GWAS. So if you want to perform GWAS, and theoretically, at least in 1 MB distance, 1 SNP should be there for any crop. That's thumb rule. But if lesser that, you have up to 100 kb or 200 kb one marker. And the best scenario is that you have a resequence, and then directly you may land up to gene, or you land up to nearby SNP and then you can again find out, explore the region to get the association. So that is what I was saying. What is LD? Then I defined it is with respect to non-random association of alleles up to a more loci in a population of individual. And that LD, understanding of LD is critical to understanding association. And in that population, what is the LD decay pattern? We must understand to know how many markers we require. So here, this particular graph is LD decay, linkage disequilibrium in a population we identified. Then how it decays, means LD, how it is LD distance block. Beyond that, there will be no LD. Means those two particular alleles, A and B, will not be together. So here you say in this particular plot, we see that LD decay distance is 1.43 MB. Means up to 1.4 MB, whatever SNP, based on that one SNP presence, you can say that other SNP is also present. So in 1.43 MB, one SNP should be present on an average to perform GWAS in the set where I have taken, like this is LD decay distance in a mini core of wheat germplasm identified in Indian National Gene Bank. So there we genotyped with 35K and here calculated this LD decay distance. We found the LD decay distance 1.43 MB, means one marker must be there to get at least region to particular region. That is reasonable level of association, GWAS association. And this LD varies depending upon what kind of individual you have taken. Like what we have taken mini core, it's very diverse. That's why LD decay distance is 1 point. We take like less diverse individual, it can be 4 MB, 5 MB, 6 MB. Then mapping resolution will go down. So you'll never end up to particular gene or particular causal SNP, which is controlling the trait. So it is always necessary that we must have high density genotyping data, or we have a very diverse collection where we can map loci at a reasonable resolution with even moderate density genotyping scale. So this is very much required. So this is a scheme. What I talked about, you need to have understanding of LD and to begin association analysis. If somebody want to perform association study in a mango, then he must first collect, identify diverse genotype of mango from different places in India, and then characterize those genotype a particular place for whatever 4 or 5 years, then characterize for the respective traits for which you want to map a gene, and then genotype them. So first requirement is a diverse set of individual. That diverse set of individual has to be genotyped, and the genotyping can be done either what I have said, you can resequence them wherever it is feasible and cost effective. Like rice, now it is 5,000 rupees. You can generate resequencing data and almost 8 to 10x per individual. And where even 50K genotyping cost is 7,000. So I never advocate 50K and 90K when resequencing can be done at a 10x coverage now at this stage, last 3-4 years. Then you can apply GBS approach, genotyping by sequencing approach in a genome where you don't have any information, a complex genome or bigger size genome where you cannot afford to generate data. Then you can just generate a little bit information. GBS is like a kind of complexity reduction approach and generating sequencing doing in a patchage. Means you don't sequence entirely, you sequence small region and align the region across the individual and discover SNP, and those SNP, basically genotyping data, is then associated with the trait. So you can do by resequencing, you can by GBS, and then you are, many people heard about 35K, 90K, 80K, whatever genotyping approach. So you, wherever it is available, those genotyping tool that can be used. In case somebody doesn't want to do resequencing or doesn't have capacity to handle those all kind of resequencing data, they can go with 50K or whatever SNP available in that particular. And then we can phenotype it. And then when genotyping file is processed, you get genotyping SNP file. Then you cannot directly go, you have to see the quality of that file and also put some filter, minor allele frequency. So for each genotype, each genotype, there will be 2 allele, like A, C. So minor allele frequency usually consider less than 5% for any marker, the marker is removed. So it is minimum threshold is 5% minor allele frequency must be there. Otherwise, it is a kind of skewed distribution of the genotype. Heterozygosity, we generally data should not have heterozygosity in case of self-pollinated crop. More the heterozygosity, it indicates your data genotyping data quality is poor. Try to reduce it, bring it up to 5 to 10%. Limit it. Don't use more heterozygosity in your data set because those may be a kind of artificial one genotype during genotyping calling hours. And missing data also should be restricted. File with 20% or more missing data should be eliminated, those markers. So then your genotyping file ready, phenotyping ready, you implement, and then you get a result in form of Manhattan plot, where you can see the marker getting associated above a significant threshold. Those markers you can select, and then further, whether is required to test them again, then those markers can be tested in bone Farani current FTR, which you will be soon in practical class. And then if you get those associated region and interested to see that whether actually they play a role in that trait, they need to be functionally validated, or their segregation in biparental population must be seen. Then only it can be said to be fully validated. So minimum requirement is co-segregation of marker in the biparental population or functional validation genome editing. You have 5-6 important genomic region, or you 2 important genome region, knock off those regions, see the effect on the phenotype. Then you'll be able to establish the functional role of the. So these are various statistical models. So you have genotyping, phenotype, all has come. You have to now apply a statistical tool and a statistical model, and that can be done using a software program. So there are various statistical tools and different software packages which can be used. So single locus model, where one locus and its association. I mean, one SNP with the trade. I mean, all the SNP effects are not simultaneously tested in a particular trade. So that is a major limiter, single. So TESL, which came the one of the initial statistical model software tool which use GLM and other things, they basically use this single locus model. So now other models have come, multi-locus model, where effect of all the SNP, like 50,000 SNP, which all the effect of all the SNP are simultaneously taken into consideration, whether some other way they are having effect on the trade. That is multi-locus, not only one locus at a time. All the locus are seen for like seed size, all the 50,000 markers, not individually seen, simultaneously seen for their effect. The algorithm design is such. And there are other models like more than two locus for more than two trades. That the multi-locus, multi-trade models have also come. So the new models are more realistic in terms of in the context of plant or organism. All the genes will be coming into picture. It's not one gene we're guiding one trade. So it's more important to test your models and more logical to apply this multi-locus or multi-locus, multi-trade model. So you have simultaneously arise 2-3 trades and all the markers are considered. So these are different. If you are interested, you can read them. These are software packages which are available. The common one are highlighted, Clean, TESL, and Gappy. Very frequent in all the paper you'll find. Nowadays, you also find Farm CPU, that is multi-locus model, another. And then MRMLM package, which also is 4, 5, or 6 multi-locus model in provided into that particular package. So that can be used. What is the limitation? Just 2 slides I'll go through. This is limitation. Most important limitation of this particular approach is the spurious association. You get many markers. So in case of single locus model, you need to correct them using Bonferroni FDR correctors. Because you can get several positive associated, you thousands of marker for one trade. So chances of getting positive marker to associate with the trade will be always high. That's why a threshold statistical context, multiple testing is used. That is Bonferroni correction and FDR is used. And other is the population status. Sometimes what happens, whatever association you get, it get because of chance, that is the population used, it basically 2 subpopulation, and trade is clear-cut divided into 2 subpopulation. I mean, there is separation in that population somewhere. So genotype A is with a small size, and genotype particular population A is associated with small size, and population B is associated with large size. So there's no all individual not considered population as suggesting that the result we are getting is not accurate. There must be all kind of haplotypes must be present and they must bear the traits. That's why here you see, here is a proper gene flow. Means you see all those genotype, 6 genotypes, you see they all are different. Here 3 are one genotype basically representing another genotype, and basically 2 genotypes representing 6 individual and saying association. That's inaccurate estimation. That's why several programs now use population structure, kinship and all. Despite that, we have the problem of population stratification. Here, one of the paper where we have conducted, we have published several papers. This is one where we have shown, we published this one in Genomics Journal, where we have used 35 line of wheat and genotype them on 35K and establish the association, find some novel candidate. And these are the picture, that Manhattan plot, LDDK plot. This is population structure. Then other aspect which we'll be dealing is the germplasm duplication identification. So just I'll take 2 minutes for this. You all know that worldwide gene banks maintain large collection. Rice, we have more than 1,20,000-30,000 collection here in India. But all 120-30 are not different. There will be many which will be actually duplicate of each other. So it is necessary to identify those, because it will otherwise incur huge cost on gene banks, because they have to divert a lot of financial resources in maintaining, evaluating, and then regenerating, and then again conserving, even exchanging, and even those who are evaluating also, they will have tough time to evaluate such a big size of germplasm. So if you identify smaller, not having redundant lines, that will be a unique set of lines which can be shared with evaluators, breeders, and they have limited resources, they can easily evaluate them for better nutritional quality, root related parameter, whether for drought tolerance, etc. So we, since genotyping approach all have come, and it is now cost effective to genotype. So we have developed a web server. It is known as germplasm identification and removal tool. So this is a kind of two-tier architecture and developed in R and uses various programs, PHP, HTML, CSS. And so here you need only and only, you don't need any phenotype. You need only genotyping data of 1 lakh rice genotype or 500 or 1000, whatever genotype you have, their genotyping data. And then you have to upload on this particular site, and there are several thresholds provided. You have to select them, and based on them, you can check the default option. You will get what are the lines they are actually duplicate of each other. So this is the last slide. So this particular practical aspect will be again taken by Tanmay Kumar. So he will be giving a practical aspect of this as well. We will try to explain how to perform Jivas and is it feasible for you people to download Tessel software. He will tell you how you can download in your computer, and if you want to perform, it will be also telling how you can perform. So thank you. If you have any query, you can ask me. And then now Dr. Tanmay will be taking the class now. So he'll be showing you how to perform Jivas and then duplicate identification using the particular server. Thank you.
Thank you so much, sir. I hope Dr. Sahu is here.
Yeah, now is here.
Okay.
So much, sir, for this elaborate presentation. Aspects. First thing we will be doing the presentation. Am I audible?
No, sir. Your volume is very low.
Mamta, whether he is audible to you?
No, sir. No.
Huh?
Not audible, sir.
Hello?
Hello.
Mamta, whether he is audible to you?
No, sir. Not audible. Hello.
Am I audible?
No,
Now,
not yet audible.
Bolo, Tanmay. No.
हेलो क्या हुआ हेलो
अब आने क्या
हेलो
हाँ मैम
Am I audible?
Yes.
can
Okay.
you raise the volume if possible?
I have raised to the maximum.
Now it is okay.
So probably it is audible right now, right?
It's very low, sir. I don't know if everybody can hear it properly. Participants, can you give your feedback if the voice is clear and audible to you?
Yes, ma'am. Audible है.
Audible है और clear है, loud है? Is it
थोड़ा
loud enough?
नेटवर्क इशू से थोड़ा प्रॉब्लम है.
Okay. So first of all, I'd like to thank Amit sir for this elaborate presentation. And I'll be taking the practical class. So first of all, we'll deal with Tassel. So you can open your tutorial, and my screen is visible to everyone. My screen is visible.
Screen is not visible. It's visible, sir. Visible.
Okay. So probably we'll be missing this link in the main tutorial file. I have added this, so I can paste it in the chat box so that you can download and install the Tassel software if you want. Did you get it? It is tassel.bitbucket.io. So from here, you can download the tassel version based on your system requirement. Like if you have a 64-bit operating system, you can download this one, tassel 5 Windows 64-bit. I have already downloaded. If you want to execute the exercise along with me, you can take 5 minutes to install. Otherwise, I will just show you, and later on you can try. So which option you want to follow?
Which option all of you want? Please reply. Would you like to install?
Okay, let me show you first. Then this is the source of the executable file I have shown you. You can download it and later on install it. So let me show uh, how we can perform the exercise in Tassel. So when you will install, you'll be getting a particular folder in your C drive under program files, and under that you'll be getting tutorial data. So initially, for learning purpose, you can use and see the data formats here. So for example, if I'll open this one, you can see how it is written, how the phenotypic file looks. So this is these are 3 variables, and under this angular bracket, trait will be mentioned, and the first column is the genotypes, and second, third, fourth contains the variables. Likewise, you can also see the genotype file that is basically given in HapMap format. Also, you can use other genotypic format like VCF format, and some other formats also exist. So here the first column deals with the SNP ID, then alleles, then chromosome number, then position, then the strand on which the particular marker is present, then the assembly, center, post ID, assay ID. All these informations will come along with the sequencing company who will be giving you. If those 5 information you don't have, you can simply put NA in place of that. Then from here onwards, your genotype name starts along with the column here. You can see. So for this genotype, as it is a text pad, so you may not get a perfect alignment.
Whether everyone is able to follow this or?
Sir, I wonder if he is performing it online, because we are able to
You
see.
want to first only go through, he will show the screenshot. So that workflow for the Tassel program. And afterward, you will download and
Directly, sir, I'll execute.
You are showing it directly?
Yeah, directly I'll show.
But it is not visible, I think. It is
Yeah, that's why.
Actually, Tanmay, it is not
showing.
visible.
My screen is visible to everyone or not?
The first one that you shared is visible. After that, it has stuck on that only. If you have performed any practical thing, so it was not at all visible.
So this file is sorry, my screen share is paused. So let me share it again. Okay. So now this file.
Yes, now it is okay.
Now it's visible.
I have to take care of this because sometimes the screen sharing is paused. Anyway, so this is the genotype file. You can see here. The first column is the SNP ID, then alleles, then chromosome number, then position, then strand. And from this part, like assembly center, prot LSID, SA LSID, if you don't have any information, then simply you can have NA in place of that. Then from this portion, portion onwards, the genotype column starts. Okay. So for example, for this SNP PZP00859, the genotype is CC for the cultivar 33-60. uh, that is the naming convention used. So before to that, I would like to show you another file. Okay. Now this one is visible to everyone. This file.
Yes.
Yes, it's visible.
So here the first column is straight in an angular bracket, and now in the first column you will be getting the name of the genotypes like 811, A6, A659, likewise B164. Then it will follow columns. I mean the number of traits you want to consider. So here they have considered 3 traits. The first one, second one, third one. Okay. So in this way, your phenotypic file should be. Okay. So now I will share the tassel interface. If you click the tassel icon from your desktop after installation, so you'll be getting this tassel interface. So there are several file you can open from here, but due to time constraint, we will follow a simple workflow which has been already shown in the tutorial. You can open the tutorial side by side. So from the workflow, they have given 4 models: MLM with Q plus K. Q is the file you will be getting from analyzing the genotypic data with structure software. That is another software. So how that file looks, that I can show you here. This one. So this file is visible.
Yes, it's visible.
Visible hai.
NDP population structure. This one. So here you'll be getting one header like covariate, and again trait will be there. Then Q1, Q2, Q3. So in this particular case, when the data was analyzed in structure, it identified the populations to be 3. Okay. So everywhere you'll be getting the membership coefficients for each genotype. For example, for this particular genotype, for Q1, this is the coefficient. For Q2, this is the coefficient. For Q3, this is, I mean, you can say the percentage of membership also. So this file will be generated from the structure software. So now we'll come back to the workflow. So from workflow, we will run the first model. So I'll show you one model. You can, go through the software later on, and you can run all 4 models. So basically what we are trying to identify, 2 things we try to identify. First thing is, what is the relation between the genotypic file and the phenotypic file? So whether that particular relationship relation is being explained by all the models perfectly, correctly. So basically, we try to identify a correct model first of all. Then our target is to get the markers from the result of that particular correct model. So if your model doesn't fit well, then you cannot, you should not proceed with that model. You should always check whether your model is fitting correctly or not. And what the parameter for that is, I mean, you can check whether your model fits correctly to the genotypic and phenotypic data by visualizing the QQ plot. First, we'll run one model and we'll check. So for the workflow, you can see here data, load genotype file, then filtration criteria, then again load population structure file, then filter traits. So you'll be getting all these things. So first of all, we will browse the genotypic file. So you can keep sort position. Sometimes the genotypic file what comes to you that is not sorted. So basically we sort the positions first. Either you can select here from HapMap or you can make best guess. So if you are doing with me, then you can create a folder in your name and you can put all the files there. So this is the MDP genotype, that is test file or tutorial file. You can open it here. So this is how the genotypic file looks. You can see in the interface of tassel. So you can color it based on your requirement also. Then the next step is filter the genotypic file. HRS said you what should be the minor allele frequency. We should keep minor allele frequency is 5%. So here you can write 0.05. So maximum allele frequency we don't bother because it should be 1 always. Minimum heterozygous portion, we should not take the minimum heterozygous portion. I mean, it should be always 0%, but the maximum should not exceed more than 10%. So here, if you want to run it in a default manner, you can keep it like that, but I would suggest you make it 0.1. We'll see what happens. Then remove minor SNP states, remove sites with indels. So indels will be removed. And minor SNP states means where you may find in some cases there are 3 alleles for a single SNP. Okay, so in that kind of SNPs will has to be removed from the data. Then you click on OK. Now your file is filtered. The next is load population structure file. So I have kept that, make best guess. Then I will browse it. Then population structure file. Okay, so this is how it looks. You can include all the 3 population like Q1, Q2, Q3. Otherwise you can selectively use also. So click on OK. We are using the file incomplete. Then next is load phenotype file. Okay, so our phenotype file is MDP traits. That's that one. This is our phenotype file. Okay, then it asks for a kinship data. The kinship means it is a file that can be generated using some workflow in the tassel also. A kinship file that gives you the relationship between the taxa, that quantifies the relationship between the taxa. So here also one kinship file is available. This one mdp.kinship. You can see here, this is the first taxa, the second taxa, and the relation between all the taxa is given. So kinship will be a matrix of the number of genotype into the number of genotype. So now with the default option, we'll be executing the MLM. So now it asks you whether you want to write the output file. Yes, we of course need the output file. So you need to browse for a particular folder. So I will give it a path in the training folder. Okay, here I'll give a path and file name. I can make like MLM. I am using a population structure. That's for I put here Q, then a kinship matrix I am using. Okay, with this name, the file will be generated. Okay, you save it. So here one option is there, filter output file on a p value. So you can mention the p value after Bonferroni correction, that I'll be letting you know. We will keep it, default. We will not check it because we will be not able to see the entire, Manhattan plot if we are putting this filter. So now our model is being executed. So are you able to follow me? Here invalid selection it is showing because it is not getting proper files. Just ignore this message because we'll be studying Manhattan plot and QQ plot from the file it has generated. So let's see whether it has generated any file or not. On the D drive, so I kept it on the training folder. So yes, it has generated some files. You can see here. Yes, this one. Is it visible? Is it visible to everyone? So now you see it has generated many files. So the most important file we should look for is MLM QK MDP genotype filter plus MDP traits.start. So that file will be giving us the main result. Okay, now what we will do, you can file open from training. We will open the starts file. Okay, now with this file we'll be getting knowing whether our model is a well fit or not. Okay, so now come to results, then QQ plot. Okay, here 3 traits are there simultaneously. For 3 traits you can also draw the QQ plot, otherwise one by one you can do. So let me select all the 3. Okay, now you see. So it is a plot between the log -10 log p, because whatever association is generated, I mean association is detected, that is based on a p value, and that p value is converted to -10 log p. Okay, so you'll be getting it here p value, and you can convert it into -10 log p. So now every model has a mathematical formula. So based on that, what should be the expected log p, and based on that, what should be the line? I mean, what should be the line? You can see this straight line is the expected line. So basically, if your model is a good fit, your trait, like you can see your HT, that follows this line up to this, then some diversion you are getting here. And the green one also follows, but the blue one is not a well fit. So there could be a possibility that the data is not very much correct, or to this data some other models can be a best fit. So for these two, dipole and ear diameter, so for these two the fitting is approximately well. So you can, if your QQ plot falls somewhere apart from this line, if it is very far from the expected line, that means your model is not a good fit. But up to a certain limit it will fit. Then after that, some markers will be apart from the main line. That means the model is a good fit, and whatever markers are not on this line, those might be associated. Okay, So this is how we can draw the QQ plot, and based on QQ plot, we can decide whether this model, whatever model we are using, that is a good fit or not. So if the model is a good fit, then we can go and visualize the Manhattan plot. So now you can select a trade. Like for this year HT, we got a good fit. You see here. So these are the markers, and the markers above a particular threshold are called as associated markers. So for example, if your value is 3, if your log p minus 10 log p value, that is the threshold is 3, then you will be getting this marker as the associated marker. But how to decide what is the actual threshold value? So as sir had explained you, for single locus models, we usually do the Born-Ferroni correction. The Born-Ferroni correction is nothing, but it is a modified p-value. So how? So the basic p-value in all the statistical analysis we consider, that is 0.05. Okay. So to that 0.05, you divide all the number of markers. The value you'll be getting, that will be the log p value. For example, in our case, we have got around 3094 markers. If we are dividing, so if you are dividing our number of markers, like 0.05 divided by 3094. So this is the p value. Okay, this is the p value threshold for us for this data. So we cannot analyze the p value like this, like e to the power -5. It is a very big extended number. So for that, you need to convert it as minus log 10p. Okay, it is, if you are taking log 10, so it is 4.79. If you will take minus log 10, so that will be like 4.79. So any marker in your case, or in this particular data set, any marker coming above this threshold should be taken. Unfortunately, for this case, we are not getting any associated markers. So similarly, we can check another another trait. For another trait, we will be checking. Okay, here you are getting something like for these markers might be above 4.79. So some markers you might be getting here. Okay, so if you are opening that particular sheet, that particular file in Excel, you'll be able to visualize whether any marker is above 4.79 or not. So that we'll be seeing here. Okay, now the tassel is visible, right? Okay, so here again I'll draw the Manhattan plot, like this particular trait will be giving. So our threshold was 4.79. So any marker, if you are visualizing here above 4.79, then you can take that marker, that edge associated marker. Okay, Similarly, for another trait also, we can see whether it is a good fit or not. Here also, you are not getting any marker because nothing, no marker is coming above 4.79. So based on your data, you'll be getting a number of markers, maybe 1, 2, 4, 5, or maybe in tens or 20s, you'll be getting some markers. So based on that, as sir explained about LD distance, so based on that, you can see how many markers are coming in a particular LD within a particular LD decay distance. That those could constitute a haplotype. And similarly, within that LD distance, if you are getting some genes associated, so that gene could be associated with the particular trait. So now I will show you this particular file in the interface of Excel to see whether we are getting any markers above 4.9. So here it is the p value. So I will put another column here. Okay, here I will type minus 10 minus log 10, then this is the p. Okay, so then put it NA because this is not a particular marker, that is log. I will put, then I will sort it. Okay, so now you see, unfortunately we could not get any marker. Up to 4 we got, 4.28 we got. If we would have got 4.79 more, some marker associated with the p minus 10 log p value more than 4.79, that marker could have been associated with the trait of interest. So you can try it with your data set. So probably we'll be getting some markers. So any doubt till this point? Okay. So now. Hello. Hello.
Yes, sir, please continue.
Okay. So now dealing with the germplasm duplicate identification.
Audible है क्या?
Yes.
Mamta, whether is audible?
Am I audible?
Yes, sir, audible, audible.
Okay.
So it is the tool that is hosted in webtools.nvpgr.ornet.in g. So you'll be getting the home page here. So something about the tool has been written here. Then the main part is the server part, the server where you need to submit the job. So here one sample HapMap file is there. I have downloaded that, and you can download this HapMap file. And if you want to continue this exercise with me also, you can do that because it is an online tool. So that file I will choose from here. This one is the file. So where one known duplicate is there, I'll be showing you where the known duplicate is present. So there are two kinds of parameters. First parameter is duplicate identification and removal parameter. So here you have two options. One is homozygous difference. Second one is total genotypic difference. That includes homozygous difference along with the heterozygous difference. Okay. The tool detects the duplicates based on identical by state analysis. So identical by state is nothing, but it is based on the allele sharing percentage. Like if one genotype at a particular locus contains a T, and in the same locus, another genotype contains a T. That means they share two alleles, like A, they share T, they share. Okay, so that is a two allele sharing locus. And another case is one is AT, another is AA. One is homozygous, another is heterozygous. There they don't share all the two alleles. They share only one allele, that is A. Okay. So that is one allele sharing. And another is no allele sharing. If both are homozygous but opposite, like one is dominant, one is recessive. One is AA, another is TT. So there will be no allele sharing. Okay. So if you consider homozygous difference, then we will say, we will just try to identify how many no allele sharing or zero allele sharing locus are present between those two genotypes. And for total genotypic difference, we consider one allele sharing and two allele sharing, both kinds of similarity. Then genotypic difference threshold 0.1%, 0.1%, it is not 10%, it is 0.1% is the default for our case, because we executed it with a data set and we got our duplicate captured within 0.1% of threshold, genotypic difference threshold. Then remove monomorphic SNP. These are the basic parameters you can select through here. Generate cluster. If you want to see the cluster of genotypes, like you have three options: rectangular, triangular, circular. You can select circular, and you can also set the resolution here. So by default we used 72 dpi as resolution. You can increase based on your requirement, because for publishing you need higher resolution figures. Then minor allele frequency, it is default 5%. Then missing data, it is 5% we have taken. You can take up to 20% if you want to allow. I mean, 5% missing data is allowed. So here you see 0.1 means 0.1%. Here 0.05 means 5%. Okay. Then missing data marker, missing data genotype. So what is the percent? I mean, here 10% missing data in genotype we allow. So linkage disequilibrium pruning. So if you want to select the diverse SNPs, so all the SNPs, those are in LD. Okay, so you can select a representative one or two, like that. So the LD threshold, you should use 0.75. Then Hardy-Weinberg equilibrium. If you are taking a particular population and you know the parents, so in that case you can use Hardy-Weinberg p-value. Otherwise, you can use 0 as no use. Then marker heterozygosity. So 0.1 means 10% we are allowing. With this, you can submit the server. It will take a little time, and it will show you the result page. So in the result come, we can see the kind of result we will be getting in the tutorial file also. Yes, it's running. So you can come to the tutorial file. Here you will be getting a kind of result you expect. So what kind of parameters? Share it again. Here the result is. So what kind of parameters you have used? Okay, summary. What is the summary? Then is there any duplicate you found, and whether you will be able to locate the duplicate in the tree you have mentioned. Then it is a K density graph based on the homozygous difference you have used. Then genotype heterozygosity will be giving you, then marker heterozygosity. So let me check whether yes, we got the result. Probably it is more clearly visible to you. So first it will show you what parameters you have used. You have used remove monomorphic SNPs true, then generate cluster, circular clusters you have used, selected as parameter, then minor allele frequency this much. Okay, all the parameter details it will be giving you. Then summary is the input file, HapMap file contains 31,926 markers for 95 genotypes. Then 95 genotypes retained after missing data filtration of 10%. Then again, for 1% homozygous difference, you are getting 94 genotypes. That means one duplicate was present. Let's locate that particular duplicate. So now LD pruning filters the markers, not the genotypes. So homozygous difference filters the genotypes, but LD pruning filters the marker. Hardy-Weinberg, heterozygous threshold, those filter the markers. Now, what is the duplicate? C306 and genotype 2 is IC128151. Then percentage of difference is 0%. So there is 0% homozygous difference. That means both are 100% identical. So if you'll see in the PGR database, you'll be getting for C306, one of the accession is registered as IC128151. So here in the picture also, tree, you can see here, if it is visible to everyone, C306 with 0.05%, 0.01% of threshold, it is pairing with IC128151. So now you'll be getting K density graph. It will show you the density of SNPs, and here you'll be getting the pairwise homozygous difference. So this is the mark where 0.1% homozygous difference has been used. Here you'll be getting genotype heterozygosity, the number of genotypes and fraction of heterozygosity you'll be getting. Here you will be getting marker heterozygous. As you have used the filter of 10% heterozygous markers. So all these markers here will be removed because they are more than 10%. So you have taken this much of markers for your analysis. So you can also download all the results file from the links provided here, high resolution images. So for detailed help, you can go to the help section. Everything is written, how to prepare your file, okay, and how the parameters are used, marker heterozygosity, homozygous difference, total genotypic difference, okay, output, then how you'll be getting. So it was a circular tree that there, that's why we could not mark the red line on that. But if you select a rectangular or triangular tree, you'll be getting a red line mark here. Then again, marker heterozygosity plot. You can also download the help manual in the form of PDF. Further, if you want to run the tool with the data sets we have used, you will be getting all the data sets we have used here. And you can download help manual from this link also, g.web server, g.offline. So if your data is very large, you can request for a R package we have developed and we are providing it on a request basis. You need to register here. Okay, then you'll be getting the offline R package. If you are well versed with R, then you'll be able to run it in your local system also. So with this, the practical is over. Any doubt, any questions, please ask me.
Thank you, Dr. Sahu. Any questions, participants? No questions. So if there are no questions, then I would like to thank our speakers of the day. And I hope that whatever was taught, though it was a short time duration to elaborate so many things, but of course it was an informative lecture, sir, and especially your work related to development of this web server will be definitely beneficial, and that if somebody is facing any problem, that help section is also there, so they can get the help from there also, and they may also contact to Dr. Sahu as well as Dr. Amit, and if they want to implement the this database for their own work, they will be happy to share and further extend their knowledge. And they said, in fact, they are asking for the YouTube link also for this lecture. May probably they are interested in doing such kind of work. So they are also asking for Dr. Sheetal Patel. She's saying that please share your number and email, as while explaining voice was low. Okay, so of course this lecture is available on YouTube also, and we will be sharing the numbers and all the information of the resource persons. And if you need, you can refer the compendium also. There will be all the information. So with this, sir, thank you so much. A special thanks to Dr. Sahu, because we also were not knowing that he'll be there. So thank you so much for providing the detailed account of TESOL, as well as the web server developed for the identification of gene duplicates. Thank you so much, sir.
Thank you. Okay.
Yes, sir. So you're saying something?
K.N. Gupta is saying something. We are not able to hear him. He is muted. Dr. K.N. Gupta is muted.
Dr. Gupta, you want to say something?
Oh, madam, thank you very much, madam. Very nice present lecture.
Okay. Thank you.
Thank you. Thank you. If you have any query, you can mail us and we'll be always ready to support you, help you. Okay. Thank you. Or you can call also if you have if you face any difficulty in running this software or anything related with this presentation or any program which was presented in this training session. Thank you, Mamta. Okay, thank you.
Thank you so much, sir, for your time. Thank you so much. So with this, today's lectures are over. This practical session is over for the day. And today again we'll meet by 9:30 to start our new session. So soon this training is going to end. So please try to attend with your full attendance, so that we also feel enthusiasm in delivering and introducing new speakers to you. So with this, I would like to urge, please be available, and thank you so much. So tomorrow we will meet by 9:30. Mandalji, if you are there, please you can end the session, sir.
Close कर दूं मैं?
हाँ जी, over हो गया है.
Okay.
हाँ जी.